Hello, friends. Welcome. I'm really glad you're here. To begin, I'd love to hear where you're watching from and what the clock says for you right now. It's amazing to see people gathering here from so many places at all sorts of hours. Thank you also to those who've subscribed and supported the channel by liking the videos. It means more than you know, and it helps others find their way here too. If you have suggestions for future topics, please share them in the comments. Now, let's begin. Chapter 1. Understanding Stem Cells and Their Power. In the vast landscape of biological discovery, stem cells emerge as remarkable entities, holding the keys to understanding life itself. Stem cells are unique in their ability to develop into many different cell types in the body. They possess two essential properties: the capacity for self-renewal, allowing them to replicate indefinitely, and the potential to differentiate into specialized cells, such as muscle, nerve, or blood cells. Imagine a single cell quietly brimming with possibility, waiting to become something extraordinary. The significance of stem cells cannot be overstated. They play a pivotal role in growth, development, and healing. As we journey through life, our bodies face constant wear and tear. Stem cells act as a repair system, replenishing lost or damaged cells, thereby maintaining our health and vitality. Consider the remarkable process of regeneration that occurs in various organisms. For example, when a newt loses a limb, it is the stem cells that gather at the site of injury, transforming into the specialized cells needed to rebuild the lost structure. This wondrous ability raises questions about the limits of healing and regeneration in humans. What if we could harness the power of stem cells to repair our own injuries or mend diseases? The historical context of stem cell research is as intricate as the cells themselves. The journey began in the mid-20th century, when scientists first isolated stem cells from mouse embryos. This groundbreaking work laid the foundation for our understanding of these extraordinary cells. In the following decades, researchers began to uncover the complexities of stem cell biology. The 1980s marked a significant milestone with the discovery of human stem cells, igniting a wave of interest in their potential applications. One of the key breakthroughs in stem cell science came in the late 20th century when scientists successfully derived embryonic stem cells from human embryos. This discovery opened new avenues for research and therapy, capturing the attention of both the scientific community and the general public. As researcher James Thompson stated, the ability to derive human embryonic stem cells is an important step toward understanding human biology and disease. With this newfound knowledge, the stage was set for further exploration. In the early 21st century, the field experienced another significant leap forward with the development of induced pluripotent stem cells, or iPSCs. By reprogramming adult cells to revert to a pluripotent state, researchers could create stem cells from a person's own tissues. This innovation not only bypassed ethical concerns surrounding embryonic stem cells, but also held the promise of personalized medicine. Imagine a world where a simple skin biopsy could yield an endless supply of stem cells tailored specifically for an individual's healing needs. As we reflect on these advancements, it is essential to recognize the dedication and perseverance of scientists who have paved the way for such discoveries. Pioneers like Shinya Yamanaka and John Gurdon, who were awarded the Nobel Prize for their work on iPSCs, transformed our understanding of developmental biology. Their research illuminated the potential of stem cells to unlock new treatments for diseases previously thought to be untreatable. The impact of stem cells extends beyond the realm of healing. They also offer insights into the fundamental processes of life and aging. By studying how stem cells behave in different environments, researchers gain valuable knowledge about what drives cellular aging and the decline of regenerative capacity. This knowledge may one day lead to new strategies for combating age-related diseases, drawing us closer to a future where the vitality of youth can be preserved. Imagine standing at the precipice of a new era in medicine, where stem cells become a cornerstone of therapeutic strategies. The potential applications are vast, ranging from regenerative treatments for spinal cord injuries to innovative approaches in treating neurodegenerative diseases like Parkinson's and Alzheimer's. The healing power of stem cells inspires hope for millions who live with chronic conditions, encouraging a belief in the possibility of recovery and renewal. As we delve into the science behind stem cells, we must also consider the ethical implications that accompany their use. The excitement surrounding stem cell research has often been accompanied by controversy, particularly regarding the use of embryonic stem cells. This complex landscape involves not just scientific inquiry, but also moral and societal dimensions. How do we balance the pursuit of knowledge and healing with ethical considerations? Engaging with these questions thoughtfully is essential as we forge ahead. In conclusion, the story of stem cells is one of hope and discovery, woven into the fabric of scientific advancement. These remarkable cells hold the potential to transform medicine, offering new ways to heal and regenerate. As we continue to explore the depths of their power, we find ourselves on the brink of revolutionary changes in our understanding of health and disease. The journey is far from over, and the future promises even greater revelations. The exploration of stem cells is not merely a scientific endeavor. It is a quest for understanding the very essence of life, healing, and the potential to transcend our biological limits. As we immerse ourselves in this realm, we encounter the notion of tissue engineering, a field that combines principles from biology, medicine, and engineering to create functional tissues and organs in the laboratory. Imagine a scenario where damaged organs are replaced with bioengineered tissues, crafted from our own stem cells. This vision is gradually becoming a reality, demonstrating the transformative power of stem cell research. Tissue engineering relies heavily on stem cells due to their unique ability to differentiate into various cell types. Scientists are experimenting with scaffolds, biocompatible structures that guide stem cells to grow and form new tissues. These scaffolds can be made from natural or synthetic materials, designed to mimic the extracellular matrix, which is the environment surrounding cells in the body. This innovative approach raises intriguing questions. What if we could create a heart or a kidney in a lab using our own cells? What if organ shortages became a thing of the past? Among the pioneers in this fascinating field is Anthony Atala, who leads the Wake Forest Institute for Regenerative Medicine. His groundbreaking work in growing human tissues and organs has brought us closer to the dream of organ regeneration. Atala states, "The body has the ability to heal itself. We just need to provide it with the right tools." This perspective emphasizes the synergy between nature and science, highlighting how stem cells can be harnessed as tools for healing. As we peer further into the potential of stem cells, we also encounter the realm of cell therapy. This innovative treatment involves introducing healthy stem cells into a patient's body to replace or repair damaged tissue. The potential applications are vast, ranging from treating blood disorders like leukemia to addressing degenerative diseases such as multiple sclerosis. Imagine a treatment where a simple infusion of stem cells could restore function and vitality to affected areas of the body. The implications of such therapies could revolutionize how we approach chronic illnesses, offering a new lease on life for many. The promise of stem cell therapy is not without its challenges, however. Researchers grapple with issues such as immune rejection, tumor formation, and the need for robust delivery methods. These challenges serve as reminders of the complexity of the human body and the intricate balance required to harness the power of stem cells effectively. As we ponder these obstacles, one cannot help but wonder, how might scientific ingenuity overcome these hurdles to unlock the full potential of stem cells? The journey of stem cell science has also illuminated the importance of understanding cellular signaling pathways. Stem cells are influenced by a myriad of signals from their environment, guiding their fate and function. This intricate web of communication is vital for maintaining proper homeostasis and ensuring that stem cells respond appropriately to the body's needs. Imagine the complexity of a symphony, where each instrument plays a role in creating a harmonious sound. In a similar manner, stem cells rely on a symphony of signals to orchestrate their behavior. Remarkable strides have been made in deciphering these signaling pathways, thanks to the efforts of researchers like Shinya Yamanaka and his team. Their work has unveiled critical insights into how extrinsic factors can influence stem cell fate, enabling researchers to manipulate these cells with precision. This knowledge opens the door to targeted therapies and personalized medicine, allowing for treatments that are finely tuned to an individual's unique biology. The historical breakthroughs in stem cell research have not only advanced our understanding of biology, but have also fostered a spirit of collaboration across disciplines. Scientists, ethicists, and policymakers are engaging in meaningful dialogues to navigate the ethical complexities surrounding stem cell research. This collaborative approach ensures that progress is not just measured in scientific achievements, but also in societal acceptance and ethical considerations. As we reflect on the trajectory of stem cell science, it becomes evident that we stand at a crossroads. The promise of regenerative medicine invites us to envision a future where healing transcends the limitations of traditional therapies. The excitement generated by recent advancements fuels a collective hope, igniting the passion of researchers and patients alike. In this ever-evolving narrative, it is essential to remain grounded in the reality that while stem cells offer profound possibilities, they also require rigorous investigation and ethical stewardship. How do we balance the drive for innovation with the responsibility to ensure safety and efficacy? Engaging thoughtfully with these questions is not just an exercise in ethics. It is an essential part of the scientific journey. As we continue our exploration of stem cells, we will delve into the various types of stem cells and their specific roles in health and disease. Understanding the nuances of these cells will deepen our appreciation for their potential and the myriad of ways they can transform medicine. Each type of stem cell, from embryonic to adult, carries its own story, contributing to the grand narrative of life and healing. In this unfolding saga, we find not only a testament to human ingenuity, but also a reminder of our connection to the natural world. Each stem cell is a thread in the intricate fabric of existence, embodying the potential for renewal and transformation. As we journey onward, let us keep our hearts and minds open to the wonders that lie ahead, for the story of stem cells is, above all, a story of hope. As we continue to explore the fascinating realm of stem cells, we encounter the various types that exist, each with its unique characteristics and roles within the body. These categories include embryonic stem cells, adult stem cells, and induced pluripotent stem cells, or iPSCs. Each type of stem cell is like a different brushstroke in a grand masterpiece, contributing to the intricate picture of human biology and health. Embryonic stem cells are the most versatile of the group. Derived from early-stage embryos, they are pluripotent, meaning they can develop into nearly any cell type in the body. This extraordinary capability places them at the forefront of regenerative medicine. Yet their use has sparked intense ethical debates. The potential benefits are immense, but so too are the ethical implications surrounding their source. As we ponder this dichotomy, we might ask, how do we reconcile the promise of these cells with the moral considerations they invoke? In contrast, adult stem cells, which can be found in various tissues throughout the body, are multipotent. They have a more limited capacity for differentiation compared to their embryonic counterparts, typically giving rise to a narrower range of cell types related to their tissue of origin. For example, hematopoietic stem cells found in bone marrow can develop into different types of blood cells. These adult stem cells play a critical role in maintaining and repairing the tissues in which they reside, acting as guardians of our bodily integrity. Then we have the revolutionary iPSCs, which have redefined our understanding of cellular potential. By reprogramming adult cells to revert to a pluripotent state, researchers have unlocked a treasure trove of possibilities. Imagine being able to take a simple skin cell, gently coax it back to a more youthful, versatile state, and then guide it to become heart muscle or neurons. This innovative approach not only opens doors to personalized medicine, but also raises profound questions about the nature of cellular identity and the power of scientific intervention. The journey of understanding these stem cell types leads to a deeper appreciation of their significance in health and disease. In scenarios of injury or illness, the body's ability to call upon these stem cells can mean the difference between recovery and long-term damage. For instance, in the case of stroke, researchers are investigating how stem cells might aid in repairing brain tissue, potentially restoring function and quality of life to those affected. This exploration is both a scientific quest and a testament to human resilience. As we immerse ourselves in the world of stem cells, we must also acknowledge the challenges that accompany their therapeutic potential. One of the most pressing issues is the risk of tumor formation. When stem cells are introduced into the body, there is a possibility that they may not differentiate as intended, leading to uncontrolled growth. This concern underscores the importance of rigorous research and monitoring. How do we ensure that the healing properties of stem cells can be harnessed without introducing new risks? Researchers are diligently working on strategies to mitigate these risks, employing advanced techniques to control stem cell behavior and enhance their safety for therapeutic use. This pursuit of knowledge exemplifies the delicate balance between innovation and caution that defines the field of regenerative medicine. Throughout this exploration of stem cells, we also recognize the importance of collaboration among scientists, ethicists, and policymakers. The path forward requires an integrated approach, where scientific advancements are accompanied by thoughtful dialogue about the ethical dimensions of such progress. This collaboration fosters an environment where breakthroughs can flourish while ensuring that ethical considerations remain at the forefront of scientific inquiry. In this spirit of shared knowledge, we can also reflect on the impact of stem cell research on our understanding of diseases. By studying how stem cells respond to various conditions, researchers are uncovering the underlying mechanisms that drive diseases like diabetes, cancer, and neurodegenerative disorders. This research not only illuminates the pathways of disease, but also paves the way for the development of targeted therapies that address the root causes rather than just the symptoms. Consider the implications of this understanding for patients who currently face chronic conditions. The hope that stem cell research brings is not just in the promise of new treatments, but also in the potential for a paradigm shift in how we approach healthcare. Imagine a world where interventions are not merely reactive but proactive, where the focus shifts from managing diseases to preventing them through regenerative strategies. As we observe the trajectory of stem cell science, it becomes clear that we are on the brink of unprecedented advances. The integration of stem cells into clinical practice could transform not only individual lives, but also the very fabric of healthcare systems. With each discovery, we inch closer to a future where healing is redefined and the boundaries of what is possible expand. The story of stem cells is multifaceted, rich with promise and challenge, and as we continue to unravel their mysteries, we find ourselves in a remarkable moment of scientific history. It invites us to consider not only what stem cells can do, but also what they reveal about our own biology and the profound interconnectedness of life. With every step we take in this exploration, we are reminded of the wonders of nature and the incredible potential that lies within each of us, a potential that science seeks to understand and harness for the betterment of humanity. Chapter 2: Types of Stem Cells, an Overview. In the rich landscape of stem cell biology, a fascinating distinction emerges between embryonic and adult stem cells. Picture the early stages of life, where a tiny cluster of cells holds the potential to become an entire organism. This is the essence of embryonic stem cells. They are derived from the inner cell mass of a blastocyst, a stage of development just days after fertilization. These cells are pluripotent, meaning they can give rise to nearly every cell type in the body. Imagine a vibrant garden where each flower can bloom into something unique, representing the vast array of possibilities that embryonic stem cells offer. In contrast, adult stem cells reside within fully developed tissues, serving as a vital repair system. These cells are multipotent, which means their differentiation potential is more limited compared to their embryonic counterparts. They are often found in specific niches, such as bone marrow or the brain, where they maintain and repair the tissue in which they reside. Visualize a skilled artisan, adept in a particular craft but not necessarily trained in others. Adult stem cells have specialized roles, maintaining the health of their native environment, but not venturing far beyond it. As we explore the depths of stem cell potential, we encounter a remarkable innovation, induced pluripotent stem cells, or iPSCs. These cells are a testament to the ingenuity of modern science. By reprogramming somatic cells, like skin cells, researchers can revert them to a pluripotent state, akin to embryonic stem cells. Imagine transforming a humble clay sculpture back into a formless lump of clay, ready to be reshaped into anything the artist desires. This breakthrough, discovered by Shinya Yamanaka, has revolutionized regenerative medicine. It allows for the creation of patient-specific cells, minimizing the risk of rejection and ethical complications associated with embryonic stem cells. The characteristics of mesenchymal and hematopoietic stem cells further enrich our understanding of this biological frontier. Mesenchymal stem cells, often isolated from bone marrow, are versatile and can differentiate into various cell types, including bone, cartilage, and fat cells. Picture a skilled craftsman capable of molding materials into furniture, decorative items, or even structural supports. Their ability to adapt allows mesenchymal stem cells to play a crucial role in tissue engineering and regenerative therapies. Hematopoietic stem cells, on the other hand, are the architects of our blood system. They are primarily located in the bone marrow and possess the unique ability to produce all types of blood cells: red blood cells, white blood cells, and platelets. Visualize a bustling factory where each worker specializes in a different task, yet all come together to create a vital product. This factory operates continuously, replenishing the blood cells that sustain life. The importance of hematopoietic stem cells is underscored by their use in treatments for various blood disorders, including leukemia and anemia. When considering the sources of stem cells, each type carries its own set of advantages and potential applications. Embryonic stem cells, with their pluripotency, present vast possibilities for understanding development and disease. However, the ethical considerations surrounding their use often cloud their promise. Adult stem cells, while limited in versatility, are invaluable for therapies that have been in practice for years, such as bone marrow transplants for cancer patients. Induced pluripotent stem cells bridge the gap between these two realms, offering a unique solution that combines the benefits of pluripotency with the ethical appeal of using a patient's own cells. The potential of iPSCs is immense, with research unfolding into applications ranging from drug development to modeling diseases. Imagine being able to create an exact replica of a patient's heart cells in a lab setting, allowing researchers to study the effects of drugs on that individual's unique biology. As we delve into the comparison of stem cell sources, it becomes clear that each type plays a pivotal role in the broader context of regenerative medicine. The choice of stem cell source often depends on the specific condition being treated. For instance, mesenchymal stem cells are being explored for their ability to repair damaged tissues in conditions like osteoarthritis, while hematopoietic stem cells are foundational in the fight against blood cancers. Consider the innovative approaches researchers are taking to harness these cells. Scientists are exploring the potential of combining different stem cell types to enhance healing. Imagine a symphony where different instruments come together to create a harmonious melody, each contributing its unique sound to produce something greater than the sum of its parts. This exploration of stem cells is not merely an academic pursuit, it is a beacon of hope for many. As researchers continue to uncover the complexities and possibilities of stem cells, the dream of regenerative therapies becomes more tangible. The stories of individuals whose lives have been transformed by these advances remind us of the profound impact stem cells can have on health and healing. The journey through the world of stem cells is a testament to the resilience of science and the endless possibilities it holds. As we continue to explore the nuances of these remarkable cells, we uncover not just the potential for healing, but also a deeper understanding of the very fabric of life itself. The narrative of stem cells is still being written, and each discovery adds a new chapter to this ongoing story. As we further explore the fascinating world of stem cells, we uncover the intricate nuances of mesenchymal and hematopoietic stem cells, two key players in the regenerative landscape. Mesenchymal stem cells, often harvested from bone marrow, have gained attention not only for their versatility, but also for their ability to exert powerful effects on other cells. They secrete a variety of signaling molecules that have the potential to modulate inflammation, promote tissue repair, and influence the behavior of neighboring cells. Imagine a nurturing gardener tending to a garden, encouraging growth and harmony among the plants through careful selection and nurturing. These cells can also differentiate into various cell types, including chondrocytes, osteoblasts, and adipocytes. This remarkable adaptability makes them a focal point in the pursuit of therapies for degenerative diseases and injuries. For instance, in the context of osteoarthritis, they are being studied for their potential to regenerate damaged cartilage, which is often a source of debilitating pain. On the other hand, hematopoietic stem cells hold the distinction of being the precursors to all blood cell types. They reside primarily in the bone marrow, but can also be found in peripheral blood and umbilical cord blood. Their primary role is to ensure a steady supply of red blood cells, white blood cells, and platelets, which are essential for oxygen transport, immune function, and clotting, respectively. Picture a bustling city where each citizen plays a vital role in maintaining order and health. The hematopoietic stem cell is the cornerstone of this system, ensuring the continuous replenishment of the blood supply. The journey of hematopoietic stem cells from the marrow to the bloodstream is a marvel of biological engineering. They undergo a series of differentiation stages, each meticulously regulated by the body's needs. When faced with an infection, for example, the body signals these cells to ramp up the production of white blood cells, enhancing our ability to combat disease. This dynamic response illustrates the remarkable adaptability of these stem cells and their critical role in maintaining homeostasis. The comparison of stem cell sources sheds light on their unique applications and challenges. Embryonic stem cells, with their broad differentiation potential, are powerful tools for understanding developmental processes and modeling diseases. However, ethical considerations often limit their use. Adult stem cells, particularly hematopoietic stem cells, have been successfully used in clinical settings for decades, especially in treating blood disorders and cancers. Their established safety profile and effectiveness make them invaluable in regenerative therapies. Induced pluripotent stem cells represent a bridge between these two realms. By reprogramming adult somatic cells to a pluripotent state, scientists have unlocked a treasure trove of possibilities. Imagine being able to create a personalized repository of cells that can be directed to become any cell type needed for treatment. This innovation not only addresses ethical concerns, but also paves the way for tailored therapies uniquely suited to each individual. As research progresses, the potential of combining stem cell types becomes increasingly appealing. The idea of using mesenchymal stem cells alongside hematopoietic stem cells, for instance, opens doors to enhanced healing mechanisms. This collaborative approach could lead to more effective treatments for complex conditions. Visualize a team of specialists, each bringing their expertise to tackle a multifaceted problem, working in harmony to maximize healing. The stories of those who have benefited from stem cell therapies remind us of the profound impact these cells can have on human health. Patients with conditions previously deemed untreatable have found hope in the promise of regenerative medicine. The journey of a child receiving a life-saving bone marrow transplant exemplifies the transformative power of hematopoietic stem cells. Through the generosity of a matched donor, life is restored and the future brightens. As we reflect on the myriad applications of stem cells, we are reminded of the delicate balance between scientific advancement and ethical considerations. The field of stem cell research is not without its challenges, but the potential rewards are immense. Each finding, each breakthrough, is a step closer to realizing the dream of regenerative therapies that can heal and rejuvenate. The exploration of stem cell types and their characteristics is a testament to the ingenuity of researchers and the boundless possibilities that lie ahead. The narrative of stem cells continues to unfold, offering glimpses into a future where healing may be as simple as harnessing the power of our own cells. As we journey deeper into this realm, the stories of hope and healing remind us that the fabric of life is woven with threads of resilience and potential. The comparison of stem cell sources reveals not just their individual merits, but also the rich interplay between their unique characteristics and therapeutic applications. Embryonic stem cells offer unmatched versatility, yet their use is often constrained by ethical debates and regulatory challenges. Adult stem cells, while more limited in their differentiation potential, have established their place in clinical practice, particularly in hematological therapies. Their reliability and safety have made them the cornerstone of treatments like bone marrow transplants, illustrating their critical role in medical history. Induced pluripotent stem cells, however, introduce a compelling narrative into this landscape. The ability to transform differentiated cells back into a pluripotent state invites us to rethink the boundaries of stem cell biology. This revolutionary approach enables the creation of personalized cell lines tailored to the individual's unique genetic makeup. Imagine the implications of developing a treatment plan based on a patient's own cells, minimizing the risk of rejection and providing a more precise therapeutic strategy. The journey of understanding stem cells also leads us to the fascinating concept of tissue engineering. This interdisciplinary field seeks to create living tissues that can replace or repair damaged organs. By harnessing the unique properties of mesenchymal and hematopoietic stem cells, researchers are exploring innovative ways to develop scaffolds that can support cell growth and tissue regeneration. Picture a cityscape under construction, where skilled architects design structures that not only serve a purpose, but also integrate seamlessly with the surrounding environment. Recent advancements have demonstrated that mesenchymal stem cells can be used to create cartilage or bone tissue in vitro, paving the way for potential treatments for conditions like osteoarthritis or fractures. This process involves carefully orchestrating the conditions under which these cells thrive, guiding them toward their desired fate. The implications for regenerative medicine are profound, offering hope for patients who face the debilitating consequences of tissue loss or damage. Hematopoietic stem cells, with their ability to continuously produce blood cells, are also being explored beyond traditional applications. Researchers are investigating their potential in gene therapy, where these cells can be genetically modified to produce therapeutic proteins or correct genetic disorders. Imagine a scenario where a single treatment could effectively cure a genetic condition by correcting the underlying defect at its source. The prospect of such therapies demonstrates the transformative power of stem cells in resolving challenges that have long confounded medicine. The future of stem cell research is not solely about advancing technology. It is also about understanding the ethical landscape that accompanies these scientific endeavors. The dialogue surrounding stem cell therapy continues to evolve, addressing concerns about sourcing, consent, and the implications of manipulating human biology. As we navigate these complex discussions, it is crucial to consider the voices of those affected by diseases and conditions that stem cell research aims to alleviate. How do we balance scientific progress with ethical responsibility? The stories of individuals who have benefited from stem cell therapies serve as powerful reminders of the human impact behind the science. Each breakthrough is not just a number in a research paper, but a life touched by renewed hope. Consider the story of a young child with a severe blood disorder receiving a hematopoietic stem cell transplant that not only saves their life, but also restores their dreams for the future. These personal narratives breathe life into the statistics, reminding us of the profound significance of our journey into the realm of stem cells. As we continue to explore the diverse types of stem cells and their potential, we are reminded that the path ahead is illuminated by both promise and responsibility. The intricate dance of science and ethics invites us to ponder the future of regenerative medicine and the role of stem cells therein. Each discovery adds a layer of understanding, revealing the deep connections between our cells, our health, and our potential for healing. In this ongoing exploration, we stand at the threshold of a new era in medicine, where the transformative power of stem cells may redefine what it means to heal. With each step forward, we inch closer to realizing the dreams of countless individuals, weaving together science, compassion, and the unyielding spirit of humanity. The narrative of stem cells is rich and complex, an unfolding story that invites us to witness the extraordinary possibilities that lie ahead. Chapter 3: Stem Cell Development, from Zygote to Specialization. The journey of stem cell differentiation unfolds like a delicate symphony, where each note contributes to a masterpiece of life. From the moment of conception, when a single zygote emerges, the potential for complexity and specialization begins. This initial cell, formed by the union of sperm and egg, is the very essence of possibility, a blank canvas destined to evolve into a magnificent organism. As the zygote undergoes division, it transforms into a morula, a ball of cells, and eventually into a blastocyst. At this stage, the inner cell mass houses embryonic stem cells, the architects of development. These cells are characterized by their pluripotency, a remarkable ability to give rise to nearly every cell type within the body. Imagine a seed nestled in rich soil, brimming with the potential to sprout into a vast array of plants. The embryonic stem cells, much like those seeds, are poised to become specialized cells, muscle, nerve, blood, and beyond. The process of differentiation begins as these stem cells respond to a variety of signals. Each cell possesses an intrinsic blueprint, guided by its genetic makeup, yet it is the external environment that steers its fate. Growth factors, signaling molecules, and the physical landscape of the developing embryo all play a crucial role in this transformative journey. How do these factors shape the destiny of a cell? Consider the way sunlight influences the growth of a plant. Just as a plant leans toward light, stem cells orient themselves in response to their surroundings, adapting and maturing into specific cell types. As embryonic development progresses through distinct stages, gastrulation, neurulation, and organogenesis, the role of stem cells becomes increasingly specialized. During gastrulation, the blastocyst reorganizes into 3 primary germ layers: ectoderm, mesoderm, and endoderm. Each layer is destined to form different tissues and organs within the body. Picture a well-orchestrated dance, where each layer takes its place, setting the stage for the intricate development of structures. The ectoderm will give rise to the skin and nervous system, the mesoderm to muscles and bones, and the endoderm to the digestive and respiratory systems. The specialization is not random, but is meticulously regulated by various genetic instructions. The genes within each stem cell are like the notes of a grand score, dictating the rhythm and melody of development. For instance, the expression of certain transcription factors can trigger a cascade of events that leads a stem cell to become a heart cell or a neuron. The intricate interplay of genes ensures that each cell knows its place and purpose in the grand design of the organism. Renowned developmental biologist John Gurdon once stated, "The nucleus of a cell contains all the information necessary to create an entire organism." This profound truth highlights the power of genetic regulation in stem cell fate determination. Specific genes must be activated or silenced at precise moments in development, directing the path from a pluripotent stem cell to a specialized cell type. The timing and coordination of these events are paramount, as missteps can lead to developmental disorders or diseases. As we contemplate the complexity of stem cell differentiation, it becomes clear that the environment plays an equally vital role. The microenvironment surrounding a stem cell, often referred to as the niche, is a dynamic and responsive entity. This niche consists of neighboring cells, extracellular matrices, and biochemical signals that collectively influence stem cell behavior. Imagine a nurturing garden where plants flourish, supported by the soil, water, and sunlight. The niche provides the necessary conditions for stem cells to thrive, guiding their fate and ensuring their proper function. In addition to the intrinsic genetic cues, external factors such as mechanical forces and chemical gradients can also dictate stem cell behavior. For example, the stiffness of the extracellular matrix can influence whether a stem cell becomes a fat cell or a bone cell. Just as a tree's roots adapt to the soil's composition, stem cells respond to the properties of their environment, adjusting their developmental trajectory accordingly. Consider the work of researchers who have manipulated these environmental factors to direct stem cell differentiation in the laboratory. By altering the composition of the culture medium or the substrate on which stem cells grow, scientists can coax them into becoming specific cell types. This ability to engineer stem cells holds tremendous promise for regenerative medicine, where the goal is to replace damaged tissues or organs. The delicate balance between nature and nurture, between genetic programming and environmental influence, is at the heart of this exciting field. As we explore the remarkable journey from zygote to specialized cell, it is vital to acknowledge the significance of each stage in embryonic development. Each moment, each signal, and each interaction shapes the destiny of the cells involved. The orchestration of stem cell differentiation is a testament to the wonders of life, a process that unfolds with incredible precision and beauty. In this tapestry of development, we also witness the emergence of adult stem cells. These cells, while possessing a more limited capacity for differentiation compared to their embryonic counterparts, play a critical role in maintaining and repairing tissues throughout life. Adult stem cells reside within specific niches in the body, constantly monitoring their environment and responding to signals of injury or stress. When a tissue is damaged, these resilient cells spring into action, differentiating into the necessary cell types to restore function. Imagine the scene of a bustling city, where skilled workers are always on standby, ready to repair infrastructure as needed. Adult stem cells act as these diligent repair workers, ensuring that the tissues they inhabit remain healthy and functional. The interplay between embryonic and adult stem cells illustrates the continuous journey of development and regeneration, reminding us that life is a constant cycle of growth and renewal. As we reflect on the complexities of stem cell development, we find ourselves pondering the profound questions of identity and purpose. What determines the fate of a stem cell? How do the intricate networks of genes and environmental signals collaborate to create the diverse range of specialized cells that constitute our bodies? The answers lie not only in the science of stem cells, but also in the very essence of life itself, a journey that intertwines biology, philosophy, and the quest for understanding. In this exploration, we are reminded of the words of scientist Bruce Lipton, who eloquently stated, "Genes are not the blueprint of life, they are the instruments of life." This perspective invites us to consider the dynamic relationship between genetics and environment, a relationship that shapes not only the development of stem cells, but also the entirety of living organisms. As we continue to unravel the mysteries of stem cell development, we embark on a path that leads us closer to understanding the potential for healing and regeneration. Each step forward in this field brings us closer to harnessing the power of stem cells for therapeutic applications, offering hope to those in need of healing and restoration. Imagining the future, we envision a world where the secrets of stem cell differentiation are fully understood, where the intricate dance of genes and environment can be orchestrated to regenerate damaged tissues and restore health. The journey from zygote to specialization is not merely a scientific endeavor. It is a profound exploration of life itself, a testament to the resilience and adaptability of living organisms. Through the lens of stem cells, we glimpse the intricate processes that govern development and healing. As we look deeper into this wondrous world, we are left with a sense of awe and curiosity, eager to uncover the next chapter in the story of life. As the journey through stem cell development unfolds, we arrive at the intricate interplay of genetic regulation that serves as a compass for cell fate. The genetic landscape within each stem cell is rich with potential, yet it is the precise orchestration of gene expression that determines whether a cell remains undifferentiated or embarks on the path to specialization. Imagine a grand conductor guiding an orchestra where each musician must follow specific cues to create a harmonious symphony. Transcription factors, the master regulators of gene expression, play a pivotal role in this process. These proteins act as molecular switches, turning genes on or off in response to internal cues and external signals. For instance, the transcription factor October4 is essential for maintaining pluripotency in embryonic stem cells. When it is expressed, the cells retain their ability to differentiate into various cell types. However, as differentiation begins, October4 levels decrease, allowing other factors to take precedence. It is a delicate balance, akin to a painter choosing the right colors to create depth and texture in a masterpiece. Consider the role of signaling pathways in this genetic regulation. These pathways are like the roads that guide stem cells on their journey, with various intersections representing decisions that lead to distinct fates. The WNT and Notch signaling pathways are prominent examples. Activation of WNT signaling can promote the development of mesodermal tissues, while Notch signaling is crucial in the formation of neural tissues. The precise timing and context of these signals are essential, as they provide the information needed for stem cells to make informed decisions about their fate. The environment in which stem cells reside provides vital cues that interact with these genetic instructions. This connection between genes and the external milieu raises intriguing questions. How do cells interpret the signals they receive? What mechanisms ensure that the right genes are activated at the right time? The answers lie in the concept of epigenetics, where chemical modifications to DNA and histones affect gene expression without altering the genetic code itself. It is as if the cells have a library of instructions, with epigenetic marks determining which pages are readable at any given moment. As stem cells differentiate, they not only adapt their genetic profiles, but also undergo profound physical changes. The transition from a pluripotent stem cell to a specialized cell type involves alterations in shape, size, and cellular architecture. For instance, consider the metamorphosis of a stem cell into a neuron. This transformation requires the development of long, branching extensions called axons and dendrites, essential for communication within the nervous system. The process is meticulously coordinated, with cytoskeletal elements reorganizing to support the new structure. At this juncture, we must also acknowledge the influence of the physical environment on stem cell behavior. The concept of mechanotransduction, the process by which cells sense and respond to mechanical stimuli, further enriches our understanding of stem cell differentiation. Cells are not passive observers. They actively engage with their surroundings. Picture a sculptor shaping clay with their hands, each touch influencing the final form. Similarly, stem cells sense the rigidity of their substrate, the flow of fluid, and the pressure exerted by neighboring cells, allowing them to fine-tune their developmental pathways. Researchers have unearthed fascinating insights into how mechanical properties of the extracellular matrix can dictate stem cell fate. For instance, a softer matrix may encourage stem cells to become neurons, while a stiffer surface can guide them toward a bone lineage. This responsiveness to physical cues exemplifies the remarkable adaptability of stem cells, highlighting the profound connection between biology and the physical world. As we traverse further into the realm of stem cells, we must consider the implications of these discoveries for regenerative medicine. The potential to harness stem cells for therapeutic purposes is immense, yet the complexity of their differentiation pathways demands careful exploration. How can we replicate the nuanced conditions of the embryonic environment in the lab? What strategies can we employ to ensure that stem cells differentiate into the desired cell types with precision? The answers may lie in the development of biomaterials that mimic the natural extracellular matrix, providing the right cues for stem cell growth and differentiation. Scientists are exploring various approaches, from designing scaffolds that promote tissue regeneration to creating hydrogels that can deliver growth factors in a controlled manner. The goal is to create a nurturing environment that not only supports stem cell survival but also guides their journey toward specialization. In this exploration, we also encounter the ethical considerations surrounding stem cell research. The potential for stem cells to revolutionize medicine brings with it a responsibility to ensure that research is conducted with integrity and respect for life. The debate surrounding the use of embryonic stem cells continues to evoke strong feelings, prompting discussions about the moral implications of manipulating life at its earliest stages. As we ponder these critical questions, we are reminded of the importance of dialogue and collaboration among scientists, ethicists, and the wider community. The path forward must balance the promise of scientific advancement with a commitment to ethical principles, fostering an environment where innovation can thrive alongside respect for human dignity. As the chapter of stem cell development unfolds, we stand at the brink of a new frontier in science and medicine. The journey from zygote to specialization is not merely a biological process. It is a profound exploration of the mechanisms that underlie life itself. Each discovery sheds light on the intricate dance between genes and environment, and as we continue to unravel these mysteries, we inch closer to realizing the full potential of stem cells in healing and regeneration. In this quest for knowledge, we must remain curious and open-minded, for the story of stem cells is still being written. Each breakthrough brings us closer to understanding the complexities of development and the possibilities that lie ahead. As we delve deeper into this captivating world, we are reminded that the journey of stem cell differentiation is a testament to the resilience of life and the boundless potential that resides within each of us. Chapter 4. Stem Cells in Regenerative Medicine, Healing and Repair. Regenerative medicine stands at the crossroads of hope and healing, a field dedicated to restoring health by harnessing the body's intrinsic ability to repair itself. The primary goal of regenerative medicine is to replace or regenerate human cells, tissues, or organs to restore normal function. This innovative approach extends beyond mere symptom management, aiming for true restoration at the cellular level. Imagine a world where damaged tissues can be revitalized, where healing happens not just through medication, but through the very cells that form the foundation of our being. At the heart of this transformative field lies the extraordinary potential of stem cells. These remarkable cells are the raw materials of our bodies, possessing the unique ability to develop into various cell types. In regenerative medicine, stem cells serve as agents of healing, playing critical roles in tissue repair and organ regeneration. They can be directed to differentiate into specialized cells, such as neurons to repair nerve damage, cardiomyocytes for heart tissue repair, or chondrocytes to restore cartilage in joints. The applications are as diverse as they are promising, painting a picture of a future where chronic conditions might be overcome through the power of our own biology. Consider the remarkable case of a patient suffering from severe spinal cord injury. In clinical trials, scientists have successfully used stem cells derived from the patient's own body to promote regeneration of damaged spinal tissues. This therapy not only aims to restore mobility, but also to alleviate pain and improve overall quality of life. The results have been promising, illustrating the profound impact stem cells can have on individuals who have lost hope. Another captivating example is the use of stem cells in treating heart disease. Heart attacks can severely damage heart tissue, leading to reduced function and increased risk of further complications. However, researchers have turned to stem cells to regenerate heart muscle. In studies, patients have received injections of stem cells into the damaged areas of the heart, resulting in improved heart function and reduced symptoms. This groundbreaking approach not only addresses the immediate damage, but also fosters long-term recovery, showcasing the potential of regenerative therapies to transform lives. The journey of stem cells in regenerative medicine is not without its challenges. As we explore these innovative treatments, we must also consider the ethical implications surrounding their use. The source of stem cells often raises complex questions. For instance, while embryonic stem cells possess vast potential, their extraction involves ethical considerations regarding the status of the embryo. In contrast, adult stem cells, which are harvested from tissues like bone marrow or fat, present fewer ethical dilemmas but are often limited in their ability to differentiate into various cell types. What is the balance between scientific advancement and ethical responsibility? This question looms large in the field of regenerative medicine. Advocates for stem cell research often emphasize the potential benefits that could arise from these therapies, while critics caution against the moral implications of manipulating human life at its earliest stages. The conversation is nuanced, and as scientists, ethicists, and policymakers engage in dialogue, the focus must remain on the welfare of patients and the integrity of scientific practice. Furthermore, the regulatory landscape surrounding stem cell therapies continues to evolve. As promising treatments emerge, regulatory bodies work to ensure that these therapies are safe, effective, and ethically developed. This dynamic interplay between innovation and regulation is essential to fostering public trust in regenerative medicine. How can we ensure that patients receive the best care while also paving the way for future breakthroughs? Open discussions about the ethical framework guiding these therapies can help navigate this complex terrain. In the realm of regenerative medicine, the stories of individuals who have benefited from stem cell therapies serve as powerful reminders of the potential that exists within us. Each case study illuminates a facet of healing, showcasing not only the scientific advancements, but also the human spirit's resilience in the face of adversity. Imagine a young athlete, sidelined by a debilitating injury, who undergoes a stem cell therapy that revitalizes their damaged cartilage, allowing them to return to their passion. These stories inspire hope, fueling the desire to explore the uncharted territories of healing. As we journey through the landscape of regenerative medicine, it becomes clear that the intersection of science and compassion is vital. The stories of researchers working tirelessly to unlock the mysteries of stem cell biology echo the age-old quest for healing. Pioneers in the field, such as Dr. Shinya Yamanaka, who was awarded the Nobel Prize for his work on induced pluripotent stem cells, remind us that the boundaries of what is possible are continually expanding. His assertion that, quote, "the ability to regenerate tissues will change the way we think about healing," encapsulates the essence of this transformative field. Imagine a future where the limitations of age and injury are but whispers of the past, where advancements in stem cell therapies can lead to not only the repair of damaged tissues, but also to the prevention of diseases that plague humanity. The potential for regenerative medicine to alter the course of our health is profound, and as we delve deeper into this subject, we discover the intricate dance of biology and innovation. The applications of stem cells in regenerative medicine are vast, each representing a unique opportunity for healing. From the potential to regenerate organs such as the liver, which has remarkable regenerative abilities in nature, to the possibility of restoring vision through retinal cell replacement, the horizon is filled with promise. Researchers are exploring how stem cells can be utilized to heal wounds, combat degenerative diseases, and even extend life itself. As we reflect on the remarkable advances in this field, we also acknowledge the importance of responsible research practices. Ethical considerations must remain at the forefront of scientific exploration. As stem cell therapies become more prevalent, it is crucial to ensure that they are developed through rigorous scientific inquiry and ethical oversight. Only then can we truly harness the power of stem cells for the benefit of all. The path of regenerative medicine, illuminated by the promise of stem cells, is a testament to human ingenuity and the pursuit of healing. Each step forward offers a glimpse into a world where the boundaries of medicine are continually redefined. As we continue this exploration, let us remain open to the possibilities that lie ahead, embracing the journey toward a future rich with healing and hope. As the promise of regenerative medicine unfolds, one cannot overlook the myriad applications of stem cells in promoting healing and repair. Each potential application represents a frontier in the quest for health restoration, a journey toward understanding how we might leverage our own biology to overcome ailments that once seemed insurmountable. The horizon glimmers with hope as we witness advances in areas such as tissue engineering, where stem cells play a pivotal role in creating new organs and repairing damaged tissues. Imagine a scenario where a bioprinter, much like an inkjet printer, utilizes stem cells as its ink to create functional tissues layer by layer. Researchers are increasingly exploring how to design such innovative bioprinted structures, which could one day replace damaged organs or tissues with precision-crafted alternatives. This concept may sound like a page from science fiction, yet it is steadily becoming a reality. The potential to fabricate tissues that mimic the natural architecture and function of human organs is at the forefront of scientific inquiry. With each advancement, we inch closer to a future where organ transplantation might become obsolete, replaced by a patient's own cells engineered to form the very organs they need. In the realm of ocular therapies, stem cells have shown astonishing promise in restoring vision to those afflicted with degenerative eye diseases. For instance, researchers have successfully used stem cells to generate retinal pigment epithelium cells, which are crucial for healthy vision. Patients suffering from conditions like macular degeneration can envision a brighter future, where their sight is not merely preserved, but rejuvenated, thanks to the regenerative capabilities of stem cells. This interplay between vision and cellular restoration embodies the heart of regenerative medicine, transforming lives through innovative solutions. Furthermore, the application of stem cells in the treatment of diabetes is a burgeoning area of research. Scientists are exploring ways to regenerate insulin-producing beta cells in the pancreas, which are damaged or destroyed in individuals with type 1 diabetes. The potential to restore normal insulin production through stem cell therapy could revolutionize diabetes management, shifting the focus from lifelong dependency on external insulin sources to a more natural, self-sustaining approach. What if the key to a life free from daily insulin injections rests within our own cells? The promise of stem cells extends beyond physical ailments. It has the potential to reshape mental health treatment as well. Recent studies have begun to explore the use of stem cells to address neurological disorders, such as Parkinson's disease and multiple sclerosis. Imagine a future where damaged neurons can be regenerated, restoring not only physical function but also the quality of life for those affected by debilitating conditions. This vision is not a distant dream. It is being pursued with fervor in laboratories around the world. As we delve into the realm of case studies exemplifying successful stem cell therapies, we find a tapestry of human stories woven together by the threads of resilience and hope. One notable case involved a patient with severe heart failure who received stem cell therapy derived from their own bone marrow. This innovative approach led to a remarkable recovery, with the patient reporting significant improvements in heart function and overall quality of life. Such narratives underscore the clinical potential of stem cell applications, illuminating the transformative power they hold. Another compelling example can be found in the realm of hematopoietic stem cell transplantation, which has become a cornerstone in treating various blood disorders, such as leukemia and lymphoma. Through this technique, patients receive healthy stem cells that can repopulate the bone marrow and restore normal blood cell production. For many, this procedure has been a lifeline, offering a chance at remission and recovery when conventional treatments have failed. The success stories that emerge from these treatments serve as beacons of hope, reminding us that the journey of healing is often intertwined with the spirit of innovation. Yet, as we embrace the potential of stem cell therapies, we must also confront the ethical considerations that accompany this powerful technology. The discussions surrounding the use of embryonic stem cells, for instance, evoke varied opinions and emotions. While the scientific community is driven by the potential to unlock new treatments, it is essential to engage in thoughtful conversations about the moral implications of sourcing these cells. The discourse is not merely academic. It reflects our values and beliefs about life, autonomy, and the future of medicine. Moreover, the rapid advancement of stem cell therapies has outpaced the regulatory frameworks that govern their use. This evolving landscape prompts us to consider how best to safeguard patient welfare while fostering innovation. The balance between encouraging scientific exploration and ensuring ethical standards is delicate yet vital. How do we create a regulatory environment that supports breakthroughs while maintaining the integrity and safety of the treatments? Engaging diverse stakeholders in this dialogue, including scientists, ethicists, and the public, can pave the way for a responsible approach to regenerative medicine. In the pursuit of understanding and harnessing the power of stem cells, we find ourselves at a remarkable intersection of science and ethics. The stories of those who have benefited from these therapies serve as a poignant reminder of the human experience at the heart of scientific inquiry. Each success story is a testament to the resilience of the human spirit and the boundless possibilities that stem cells represent. As we continue to explore the vast landscape of regenerative medicine, let us remain mindful of the ethical dimensions that accompany our discoveries. The journey forward is not only about advancing technology, it is also about nurturing the compassion and responsibility that drive our quest for healing. Each step taken in this field can illuminate new pathways toward health and restoration, fostering a future where the promise of stem cells is realized in ways we are only beginning to imagine. Chapter 5. Stem Cells and Disease. Understanding Pathology. In the realm of medical science, stem cells serve as a bridge to understanding the intricate complexities of disease. Their unique properties allow researchers to model diseases, providing insights into the mechanisms that drive various conditions. Visualize a laboratory brimming with petri dishes and microscopes, where scientists meticulously cultivate stem cells, coaxing them into reproducing the very nuances of human disease. This process is akin to creating a living manuscript of pathology, one that can reveal the hidden narratives of health and illness. As researchers delve into the world of cancer, they uncover the enigmatic nature of cancer stem cells. These cells are not merely byproducts of tumor growth. They are the architects of malignancy. Imagine a garden where some plants flourish while others wither, yet the roots remain robust and tenacious. Cancer stem cells operate in a similar fashion, maintaining the growth and resilience of tumors. Dr. John Dick, a pioneer in cancer stem cell research, elucidated this concept when he stated that cancer stem cells are the seed of the tumor. This profound insight emphasizes how targeting these cells could be pivotal in eradicating cancer at its source. Investigating cancer stem cells involves identifying their unique markers and understanding their behaviors. By studying these cells, scientists are not only unveiling the mechanisms of tumor growth, but also discovering the ways in which these cells resist conventional therapies. This resistance raises critical questions. What if we could tailor treatments to specifically target these resilient cells? What new therapies could arise from understanding the biology of cancer stem cells? The answers lie in the integration of stem cell research with therapeutic strategies, potentially leading to breakthroughs in personalized medicine. The potential of stem cells extends beyond oncology, reaching into the realm of genetic disorders. Imagine a world where inherited diseases, once deemed unmanageable, can be studied and treated at their roots. Stem cells provide a powerful tool for this endeavor, allowing scientists to model genetic disorders in ways previously thought impossible. For instance, researchers have successfully derived stem cells from patients with conditions such as cystic fibrosis or sickle cell anemia. These cells carry the genetic blueprint of the individual, enabling scientists to explore the cellular consequences of these mutations. Picture a laboratory where a scientist gazes into a microscope, observing how a single genetic alteration can lead to a cascade of cellular dysfunction. By studying these cells, researchers can gain a deeper understanding of disease mechanisms, offering the potential to develop targeted therapies that correct the underlying genetic defects. This approach echoes the sentiment of Dr. Shinya Yamanaka, who remarked that induced pluripotent stem cells enable us to create patient-specific models for studying disease. The implications of this work are profound, paving the way for innovative treatments that could transform the lives of those affected by genetic disorders. The horizon of personalized medicine shines brightly with the promise that stem cell research brings. Imagine a future where treatments are as unique as the individuals receiving them. By utilizing stem cells derived from a patient's own tissues, researchers can create bespoke therapies that are tailored to the specific genetic and molecular landscape of each individual. This concept of personalized medicine is not just a dream. It is becoming a reality as scientists harness the potential of stem cells to develop targeted interventions. The process begins with the collection of stem cells, which can be sourced from various tissues, including skin or blood. Once isolated, these cells can be reprogrammed into induced pluripotent stem cells, granting them the ability to differentiate into any cell type. Consider the possibilities. A patient suffering from Parkinson's disease could have their own skin cells transformed into neurons, allowing researchers to study the mechanisms of the disease in a way that reflects the patient's unique biology. This approach is not merely about creating models, it's about crafting pathways to innovative treatments that resonate with the intricacies of human health. As stem cell research continues to unfold, the importance of ethical considerations becomes paramount. The journey from the laboratory to the clinic must be navigated with care, ensuring that the advancements in science align with the values of society. Conversations around the ethical implications of stem cell use are essential as they shape the landscape of research and therapy. Dr. Francis Collins once emphasized the need for ethical foresight, stating, "We have a responsibility to ensure that science serves humanity." Thus, the path of discovery in stem cell research is not solely one of scientific exploration. It is a journey that intertwines innovation with an unwavering commitment to ethical responsibility. As we ponder the future of medicine, we must consider how these remarkable cells will not only enhance our understanding of disease, but also guide us toward a future where healing is personalized, informed, and compassionate. The interplay between stem cells and disease modeling opens up new avenues for understanding the complexities of human health. As we explore the depths of cellular behavior, we uncover the stories that lie beneath the surface of illness. Imagine the potential to halt a disease in its tracks, to intervene at the cellular level before the symptoms even manifest. This vision, once distant, is inching closer to reality through the lens of stem cell research. In the vast landscape of knowledge, stem cells emerge as both a beacon of hope and a cornerstone of medical science. They invite us to dream of a world where diseases are not simply endured but understood and ultimately conquered. The challenges are significant, yet the potential rewards are immeasurable. Each discovery brings us closer to a future where the mysteries of disease are unveiled and the healing power of stem cells is fully realized. The journey is ongoing, a testament to human ingenuity and the relentless pursuit of knowledge that defines our species. As researchers continue to peel back the layers of understanding surrounding stem cells, one of the most compelling areas of investigation is the role of stem cells in modeling diseases. This innovative approach allows for a closer examination of the pathological processes at play within various conditions. Imagine a scientist carefully recreating the environment of a specific disease within a laboratory setting, a living representation of the human experience, where every cellular response can be monitored and analyzed. Within these models, scientists can observe the intricate interactions that take place at the cellular level. For instance, in diseases like diabetes, stem cells can be instrumental in revealing how insulin-producing cells fail over time. Visualize a delicate balance, where the harmonious function of these cells is disrupted, leading to a cascade of complications. By studying stem cells derived from diabetic patients, researchers can gain insights into the molecular changes that occur, illuminating the path toward potential therapies that restore balance to this system. The implications of such research extend far beyond the confines of the laboratory. By leveraging stem cells to create disease models, scientists can test new drugs in environments that closely mimic human physiology. This approach not only streamlines the drug development process but also enhances the likelihood of discovering effective treatments. It raises the question, how can we harness the stories told by these cells to rewrite the narratives of disease? Each experiment, each observation, contributes a verse to the ongoing saga of healing. A particularly poignant area of focus lies in cancer research, where the investigation of cancer stem cells reveals a deeper understanding of tumor biology. These cells, often resistant to traditional therapies, can regenerate tumors even after extensive treatment, making them a formidable challenge in oncology. By modeling cancer stem cells, researchers can study their unique properties, seeking to uncover the vulnerabilities that might allow for targeted therapies. Imagine the thrill of discovery as scientists unearth the specific pathways that govern the survival of these cells, potentially leading to innovative strategies that can dismantle tumors from within. One of the most inspiring figures in this field is Dr. Sidney Farber, known as the father of modern chemotherapy. His dedication to understanding cancer and its treatment paved the way for the development of targeted therapies that have saved countless lives. He once stated, "The best way to predict the future is to create it." This sentiment resonates deeply within the realm of stem cell research, as scientists strive to shape a future where cancer can be more effectively managed and treated. Beyond cancer, the exploration of genetic disorders through the lens of stem cells opens new pathways for understanding and intervention. Consider the potential of using patient-derived stem cells to model conditions such as Huntington's disease or muscular dystrophy. By examining the cellular consequences of specific genetic mutations, researchers can paint a clearer picture of how these disorders manifest at the cellular level. Imagine a bridge being built between the genetic blueprint and the physical symptoms, allowing for a comprehensive understanding of the disease. The use of stem cells in studying genetic disorders carries profound implications for developing therapies. By identifying the cellular pathways disrupted by mutations, scientists can explore potential corrective measures. This pursuit is not merely academic. It holds the promise of real-world impact on the lives of those affected by genetic conditions. It begs the question, could we one day enable cells to correct their own errors, restoring normal function? The potential for gene editing technologies, such as CRISPR, to be integrated with stem cell research could revolutionize our approach to these disorders. As we envision the future of medicine, the concept of personalized treatments tailored to individual genetic profiles becomes increasingly viable. Picture a world where treatments are designed not just for the disease, but for the unique genetic makeup of each patient. This vision is grounded in the advancements made through stem cell research, as scientists work to develop therapies that resonate with the intricacies of human biology. In this landscape, the role of stem cells extends far beyond their inherent properties. They serve as conduits for understanding disease mechanisms, modeling conditions, and ultimately guiding therapeutic interventions. The integration of stem cell research with emerging technologies offers a glimpse into a transformative era of medicine, one where healing is a deeply personal experience. The path forward is not without its challenges. Ethical considerations will continue to play a significant role in shaping the direction of stem cell research. As we navigate the complexities of scientific advancement, it is essential to foster dialogue around the implications of our discoveries. The voices of ethicists, patients, and researchers must intertwine, ensuring that the trajectory of stem cell research aligns with societal values and aspirations. Through thoughtful discourse and collaboration, we can cultivate an environment where the promise of stem cells is realized responsibly. Each breakthrough in this field reinforces the notion that our journey is not just about scientific achievement, but about enhancing the human experience. The narratives of disease, once shrouded in mystery, can be rewritten through the lens of stem cells, bringing hope and healing to countless individuals. As we look to the future, the potential for stem cells to revolutionize our understanding of disease is profound. They invite us to dream of a world where interventions are grounded in the very essence of our biology. The journey continues, with each discovery illuminating the path ahead, guiding us toward a future where health is not merely the absence of disease, but a dynamic state of well-being, crafted by the remarkable capabilities of stem cells. Chapter 6. Techniques for Stem Cell Research and Application. The journey of stem cell research and application unfolds through a series of intricate techniques, each designed to harness the extraordinary potential of these remarkable cells. At the core of this exploration lies the art of isolating and culturing stem cells, a process that transforms theoretical possibilities into tangible realities. Imagine a serene laboratory, where dedicated scientists work diligently, surrounded by glass containers filled with nutrient-rich media, nurturing stem cells as they grow and multiply. Isolation techniques vary, tailored to the source of the stem cells. For instance, embryonic stem cells are extracted from the inner cell mass of a blastocyst, a stage of development just days after fertilization. This delicate process requires precision, as the cells must be harvested without causing harm to the developing embryo. On the other hand, adult stem cells can be isolated from tissues like bone marrow or adipose tissue. Here, the challenge lies in the need to carefully extract these cells from their native environments, where they reside amidst a complex web of supportive cells. Culturing these isolated stem cells is crucial for their subsequent applications. Scientists provide ideal conditions, often mimicking the natural environment to encourage self-renewal and differentiation. The use of specialized media, growth factors, and extracellular matrix components creates a nurturing habitat, allowing the cells to thrive. Picture these cells as tiny seeds, each one poised to sprout into a unique form of life, given the right conditions and guidance. As the field progresses, advancements in gene editing technologies have become pivotal in the realm of stem cell research. One of the most significant breakthroughs is CRISPR, a powerful tool that allows for precise editing of genetic material. This technology has revolutionized our ability to manipulate the genome, enabling researchers to correct genetic defects or enhance specific traits in stem cells. Imagine the possibilities that arise from this capability. Scientists can now envision creating stem cells that are not only capable of differentiating into desired cell types, but are also free from genetic predispositions to certain diseases. The implications of gene editing extend far beyond the laboratory. Consider the potential to engineer stem cells that can resist the ravages of aging or those that can regenerate damaged tissues more effectively. Researchers are exploring the application of CRISPR in various contexts, from developing therapies for genetic disorders to enhancing the regenerative capacities of stem cells. This innovative technology allows us to reimagine the future of medicine, where treatments are tailored to individual genetic profiles, maximizing efficacy and minimizing risks. However, the journey of stem cells does not end with isolation and editing. Assessing their potency and differentiation capabilities is a critical step that ensures their effective application in therapies. Stem cell potency refers to the ability of a stem cell to differentiate into various cell types. Scientists employ a range of assays to evaluate this aspect, using techniques such as flow cytometry and differentiation assays to determine the expression of specific markers associated with different cell lineages. Imagine a palette of colors, each hue representing a different cell type. The challenge lies in ensuring that the stem cells can transition from the blank canvas of pluripotency to the vibrant spectrum of specialized functions. Moreover, the process of differentiation is guided by both intrinsic and extrinsic factors. Stem cells respond to a multitude of signals from their environment, adapting their fate accordingly. Growth factors, cytokines, and physical cues from neighboring cells orchestrate this complex symphony of change. As researchers delve deeper into the nuances of differentiation, they uncover the intricate pathways that govern cell fate decisions. The elegance of this process reflects the remarkable adaptability of stem cells, making them ideal candidates for regenerative medicine. Yet the path to harnessing stem cells is fraught with challenges, particularly concerning their preservation and transportation. The delicate nature of these cells requires meticulous handling to maintain their viability and functionality. Cryopreservation is a common technique employed to store stem cells long-term, enabling researchers to pause their development and resurrect them when needed. This process involves cooling the cells to sub-zero temperatures, effectively halting all metabolic activity. Imagine a serene winter landscape, where everything is frozen in time, awaiting the warmth of spring to awaken life once more. Transportation of stem cells poses another layer of complexity. When cells are shipped between laboratories or clinical settings, maintaining the appropriate temperature and conditions is paramount. Any deviation can lead to diminished viability or altered functionality, jeopardizing the research or therapy at hand. Scientists devise innovative packaging solutions, incorporating temperature-controlled systems and protective media to safeguard these precious cells during transit. The careful orchestration of these logistics ensures that stem cells can seamlessly move from one environment to another, ready to fulfill their potential in healing and regeneration. As we reflect on the techniques that underpin stem cell research and application, we begin to appreciate the delicate balance of science and artistry involved. Each step requires not only technical expertise, but also a deep understanding of the biological intricacies that govern stem cell behavior. The vision of a future where stem cells are routinely employed in clinical settings feels increasingly attainable, fueled by the relentless pursuit of knowledge and innovation. In the hands of skilled scientists, stem cells are not merely biological entities. They are embodiments of hope, potential, and the promise of healing. As we continue this exploration, we find ourselves at the intersection of discovery and application, where the techniques of today pave the way for the therapies of tomorrow. What stories will these remarkable cells tell as we deepen our understanding of their capabilities? What new horizons await us in the ever-expanding realm of stem cell research? The answers lie just beyond the horizon, shimmering with possibility. The complexity of stem cell research extends beyond the initial steps of isolation and culturing. After successfully nurturing these cells, the next crucial phase involves assessing their potency and differentiation. Potency, the ability to transform into various cell types, is a defining characteristic of stem cells, especially when considering their therapeutic applications. Imagine the thrill of discovery as researchers employ a range of assays to explore this potential, revealing the hidden capabilities of these remarkable cells. Flow cytometry, a sophisticated technique that uses lasers to analyze cell populations, allows scientists to examine the expression of surface markers associated with different lineages. This method serves as a window into the identity of stem cells, offering insights into their readiness to differentiate. Picture a vibrant mosaic of colors illuminating the laboratory, each hue representing a unique cell type emerging from the pluripotent state. The challenge lies in ensuring that these stem cells can not only retain their identity, but also transition smoothly into specialized forms. Differentiation, the process by which stem cells develop into distinct cell types, is both a science and an art. It is influenced by a diverse array of factors that guide the cell's fate. Growth factors, signaling molecules, and interactions with neighboring cells create a dynamic environment, akin to a conductor leading an orchestra. Each component plays its part in this intricate symphony, shaping the destiny of stem cells. Researchers strive to decode this symphony, unraveling the pathways that dictate whether a stem cell becomes a neuron, a cardiac cell, or another specialized type. As we contemplate the elegance of differentiation, it is essential to recognize the pioneering work of scientists who have illuminated these pathways. One such luminary, Shinya Yamanaka, famously stated that the ability to reprogram somatic cells into induced pluripotent stem cells has opened a new avenue for regenerative medicine. This groundbreaking work not only deepens our understanding of differentiation, but also highlights the potential to create patient-specific stem cells for therapeutic purposes. Imagine the profound impact of such advancements, tailoring treatments to individual needs, fostering a new era of personalized medicine. However, the journey from stem cell to specialized cell is not without its hurdles. One major challenge lies in ensuring that cells differentiate in a controlled and predictable manner. The quest for consistency in differentiation protocols is ongoing, as researchers strive to refine their techniques and optimize conditions. What if we could design the perfect environment for stem cells, one that facilitates their natural inclination toward specialization? The answer may lie in the integration of biomaterials and tissue engineering, combining the principles of biology with the innovation of engineering to create scaffolds that provide structural and biochemical cues. In parallel, the preservation and transportation of stem cells present another set of complexities that cannot be overlooked. As we delve further into this realm, it is crucial to understand the delicate nature of these cells. Cryopreservation is a widely adopted technique for long-term storage, where cells are cooled to sub-zero temperatures, entering a state of suspended animation. This process, while effective, requires precise protocols to prevent ice crystal formation, which can damage cellular structures. Imagine a tranquil scene where cells lie in a state of slumber, awaiting the moment they are called back to life, ready to fulfill their purpose. The transportation of stem cells adds an additional layer of intricacy to their handling. When scientists ship these precious cargoes across the globe, maintaining optimal conditions is paramount. Temperature fluctuations or improper handling can compromise cell viability, undermining the extensive work that has gone into their cultivation. To safeguard these cells, innovative packaging solutions have emerged, incorporating temperature-controlled systems and protective media. Picture a carefully orchestrated ballet of logistics, where the journey of stem cells is meticulously planned, ensuring they arrive safely to their destination, ready to contribute to groundbreaking research or therapies. As we reflect on the technological advancements in gene editing such as CRISPR, it is essential to consider their implications on the preservation and transportation of stem cells. With capabilities to edit genes with unprecedented precision, researchers can enhance the resilience of stem cells during storage and transport. Imagine a world where stem cells are not only edited for therapeutic potential, but are also fortified against the challenges of preservation. This duality of enhancement and protection is a tantalizing prospect, opening new avenues for the future of stem cell applications. The landscape of stem cell research is continually evolving, driven by the relentless pursuit of knowledge and innovation. Each technique, from isolation to assessment, preservation, and transportation, contributes to the broader narrative of healing and regeneration. The challenges faced by scientists are not merely obstacles. They are opportunities for creativity and discovery, pushing the boundaries of what is possible in regenerative medicine. As we explore these intricate techniques, we find ourselves on the brink of a new frontier. The stories of stem cells are woven into the fabric of our understanding of biology, medicine, and the potential for renewal. What new revelations await us as we continue to unveil the secrets of these remarkable cells? With each discovery, we inch closer to unlocking the full potential of stem cells, illuminating a path toward a future rich with promise and possibility. Chapter 7. Stem Cells in Clinical Trials, Progress and Challenges. In the realm of medical innovation, clinical trials serve as the vital bridge connecting the promise of scientific discovery to real-world applications. As we look to the horizon of stem cell research, numerous clinical trials are underway, each a testament to humanity's relentless pursuit of healing and regeneration. These trials span a variety of conditions, from neurodegenerative diseases to orthopedic injuries, illuminating the path toward potential therapies that could change the lives of millions. Imagine a world where children with spinal cord injuries can walk again, or where Alzheimer's patients can regain lost memories. These dreams are not as far-fetched as they may seem, thanks to the ongoing exploration of stem cell therapies in clinical settings. For instance, one exciting area of study focuses on the use of neural stem cells to treat conditions such as Parkinson's disease. These stem cells have the ability to differentiate into neurons, potentially replacing those that have been lost to degeneration. The hope is that, by introducing healthy neural cells into the brain, we can rejuvenate its function and restore lost capabilities. Success stories from recent studies illuminate the remarkable potential of these therapies. Take, for example, the clinical trials investigating the use of stem cells derived from bone marrow to treat heart disease. Patients suffering from severe cardiac conditions have been treated with these stem cells, leading to improvements in heart function and quality of life. Researchers have reported not only enhanced cardiac performance, but also increased exercise capacity and reduced symptoms of heart failure. Such outcomes not only bring hope to individuals facing dire diagnoses, but also inspire further research into the mechanisms underlying these benefits. As we marvel at these advancements, it is essential to recognize the regulatory landscape governing stem cell therapies. The journey from laboratory bench to bedside is fraught with scrutiny, as regulatory bodies strive to ensure both the safety and efficacy of these treatments. In many countries, stem cell therapies are subject to rigorous clinical trial protocols designed to assess their potential risks and benefits. The United States, for example, relies on the Food and Drug Administration to oversee the approval process for new therapies. This process includes preclinical studies, which provide foundational data before moving into human trials. The challenge lies in balancing rapid innovation with patient safety. Regulators must consider the ethical implications of stem cell research, especially when it involves embryonic stem cells or the manipulation of gene expression. The voices of ethicists and scientists alike contribute to this dialogue, as they navigate the delicate balance between advancing science and respecting human dignity. As Dr. Francis Collins once stated, science and faith can coexist harmoniously. This view encourages a thoughtful discussion on the moral dimensions of stem cell research, ensuring that progress does not come at the expense of ethical considerations. Despite the encouraging progress, translating research findings into clinical practice remains a complex challenge. Many promising therapies exist in the laboratory stage, yet only a fraction successfully transition to clinical trials. One major hurdle is the variability of stem cell sources. Different types of stem cells may behave unpredictably when introduced into the human body. For instance, while embryonic stem cells offer vast potential due to their pluripotency, they also pose risks of tumor formation. Meanwhile, adult stem cells, although more stable, come with limitations in their differentiation capabilities. Moreover, the collection and manipulation of stem cells can introduce additional challenges. The process of isolating stem cells requires precision and expertise, as any misstep can affect their functionality. Researchers are continually refining techniques to enhance the yield and purity of stem cells, ensuring that the cells used in trials are of the highest quality. Imagine the painstaking work of a skilled artisan, meticulously shaping a masterpiece from raw materials. Each refinement in technique brings us closer to realizing the full potential of stem cell therapies. As clinical trials progress, the importance of patient engagement becomes increasingly evident. Patients are not merely subjects, they are partners in the journey of discovery. Their insights and experiences can guide researchers in tailoring therapies to meet real needs. Involving patients in the design and implementation of clinical trials fosters a sense of community and purpose, enriching the research process. It reminds us that behind each data point is a person whose life may be transformed by a successful therapy. In the face of these challenges, stories of resilience and determination emerge. Consider the case of a young man paralyzed from the waist down due to an accident. He became a participant in a clinical trial exploring the use of stem cells to regenerate nerve connections. His journey, filled with hope and uncertainty, is emblematic of the countless individuals impacted by these trials. Through his eyes, we witness the profound impact that stem cell research can have, not only on physical health, but also on emotional well-being. As we reflect on the progress made in clinical trials involving stem cells, we are reminded of the myriad possibilities that lie ahead. Each trial represents a step toward unlocking the secrets of regeneration and healing. The road may be fraught with obstacles, but the collective efforts of scientists, clinicians, and patients illuminate a path forward. Ultimately, the quest for effective stem cell therapies is not just a scientific endeavor, it is a deeply human one. It speaks to our innate desire to heal, to recover, and to thrive. As the stories of success and struggle unfold, they weave a narrative of hope that resonates across disciplines and communities. The future of stem cell therapy is not simply a matter of scientific progress. It is a testament to what we can achieve when we come together in pursuit of a common goal, restoring health and improving lives. In this unfolding narrative, we continue to ask ourselves, how can we harness this extraordinary potential? What new horizons await as we deepen our understanding of stem cells and their applications? Each question leads us closer to a future where the remarkable capabilities of stem cells might not only be realized, but celebrated. Current clinical trials involving stem cells reflect a burgeoning field of study that is continually evolving and expanding. Researchers are exploring a wide spectrum of conditions, such as diabetes, multiple sclerosis, and even age-related macular degeneration. Each trial serves as a conduit for innovation, where hypotheses are tested against the backdrop of human health. Picture a vast sea of potential, with each trial as a vessel, setting sail toward uncharted waters of medical possibility. Among the success stories, the application of induced pluripotent stem cells, or iPSCs, stands out. These cells, derived from adult somatic cells, retain the ability to differentiate into nearly any cell type, reminiscent of their embryonic counterparts. The groundbreaking achievement in this area was awarded to Shinya Yamanaka, who stated, "All cells are the same, but they are not." His research opened the door to reprogramming adult cells, allowing them to revert to a pluripotent state. Imagine the implications of such a discovery, where skin cells can be transformed into neurons, potentially providing new avenues for treating neurodegenerative diseases. In another illuminating trial, researchers investigated the use of stem cells to treat type 1 diabetes by encapsulating insulin-producing pancreatic cells. This approach not only aimed to restore insulin production, but also sought to retrain the immune system to prevent the attack on these cells. Here lies a fascinating question. Could we one day teach the body to heal itself? The promise of regenerative therapies points toward a future where chronic conditions could be managed through cellular restoration rather than lifelong medication. As these clinical trials gain momentum, the regulatory landscape remains a critical factor influencing their success. Regulatory bodies across the globe are tasked with ensuring that stem cell therapies are both safe and effective before they can enter the mainstream. The nuances of this process reflect a commitment to patient safety, yet they also pose challenges for researchers eager to bring their findings to clinical practice. The European Medicines Agency and the FDA each have their own frameworks guiding the approval of stem cell therapies. For instance, the complexity involved in classifying stem cells as either advanced therapy medicinal products or human tissues can significantly impact the pathway to approval. Such distinctions can determine whether a therapy is subjected to rigorous clinical trials or can proceed to market more swiftly. This regulatory maze can sometimes stifle innovation, as researchers grapple with navigating the rules while ensuring the scientific rigor needed to satisfy regulatory demands. Moreover, as stem cell therapies evolve, so too does the ethical discourse surrounding them. Ethical considerations are not just an afterthought. They are integral to the dialogue among scientists, regulators, and the public. Conversations about consent, particularly regarding the use of embryonic stem cells, remain at the forefront of this discourse. These discussions prompt us to reflect on the ethical boundaries of scientific exploration. How do we reconcile the thirst for knowledge with the moral implications of our choices? The challenges of translating research into clinical application extend beyond regulatory hurdles. One significant obstacle is the variability in patient responses to stem cell treatments. Factors such as age, genetic background, and the presence of comorbidities can influence outcomes in unpredictable ways. This variability raises a poignant question: how can we ensure that treatments are tailored to meet the diverse needs of individual patients? The answer lies in harnessing advances in personalized medicine, where genomic information can guide therapy selection and improve efficacy. In recent years, the advent of CRISPR technology has emerged as a beacon of hope in this landscape. This revolutionary gene editing tool has the potential to correct genetic defects at their source. Imagine the power of editing a faulty gene that leads to disease, like a skilled editor refining a manuscript to enhance its clarity. The integration of CRISPR with stem cell therapies could pave the way for personalized treatments that not only address symptoms but also tackle the root causes of diseases. Alongside these advancements, the importance of ongoing education for both researchers and the public cannot be overstated. As stem cell research continues to progress, fostering a well-informed community will be crucial for navigating the ethical and regulatory complexities of the field. Public understanding can shape perceptions, influence policy, and ultimately support the responsible advancement of stem cell therapies. In the face of these multifaceted challenges, stories of hope and determination continue to unfold. Consider the inspiring journey of a woman who participated in a clinical trial for a rare genetic disorder. After years of searching for answers, she found herself among a group of individuals whose lives were transformed by a novel stem cell therapy. Her voice, echoing with gratitude and resilience, serves as a reminder of the profound impact that these trials can have on the human experience. As we immerse ourselves in the ongoing narrative of stem cell clinical trials, we discover a vibrant landscape filled with promise and potential. Each success story serves as a stepping stone toward a future where regenerative medicine may no longer be a distant dream, but a tangible reality for many. The journey has just begun, and the possibilities stretch infinitely before us. What awaits us on this path? The next chapter in stem cell research promises to be filled with groundbreaking discoveries, innovative therapies, and a deeper understanding of the human body's capacity for healing. Each question we ponder serves as fuel for exploration, propelling us forward into a future illuminated by the promise of stem cell therapies. The horizon beckons, inviting us to continue our quest for knowledge and healing, guided by the stories of those who inspire us to push the boundaries of what is possible. Chapter 8: Ethical Considerations in Stem Cell Research. In the intricate world of stem cell research, ethical considerations stand as a pivotal element in shaping the future of this promising field. The debates surrounding the use of embryonic stem cells often ignite passionate discussions. These cells, derived from early-stage embryos, offer unparalleled potential for regenerative therapies. Yet they also raise profound ethical questions about the moral status of the embryo. Should we consider these early forms of life as mere biological material, or do they embody the essence of human potential? The ethical discourse does not merely dwell in philosophical realms, but resonates deeply within the hearts and minds of individuals and communities. Many argue that the ability to harness embryonic stem cells for groundbreaking treatments justifies their use. Imagine the possibilities: repairing spinal cord injuries, treating degenerative diseases, or alleviating the burden of conditions that currently have no cure. Yet, opposing voices highlight the sanctity of life, emphasizing that each embryo represents a potential human being, deserving of respect and dignity. This dichotomy presents a compelling challenge. How do we honor the potential for healing while respecting the moral implications of our choices? Informed consent and patient rights further complicate the landscape of stem cell research. As we venture into this realm, it is essential that participants in research studies are fully aware of the implications of their involvement. Imagine being asked to contribute to a study that holds the promise of revolutionary treatments for diseases that afflict your loved ones. You would want to understand not only the benefits, but also the risks and ethical considerations involved. Researchers have the responsibility to ensure that participants are provided with comprehensive information, allowing them to make informed decisions. Moreover, the issue of consent extends beyond individual participation. It encompasses broader societal implications, particularly as we consider the diverse perspectives of different communities. For instance, how do cultural beliefs and values influence perceptions of stem cell research? In some cultures, the sanctity of life may lead to a more stringent stance against the use of embryonic stem cells. In others, the potential for healing may take precedence. This rich tapestry of beliefs creates a complex dialogue, urging researchers and policymakers to approach ethical considerations with sensitivity and respect. Balancing innovation with ethical responsibilities forms the heartbeat of stem cell research. The quest for new therapies is often accompanied by a sense of urgency, driven by the desire to alleviate suffering and enhance quality of life. However, innovation should never come at the expense of ethical integrity. The responsibility lies with researchers, institutions, and regulatory bodies to create frameworks that prioritize ethical considerations. Imagine a world where innovation and ethics coexist harmoniously, where groundbreaking therapies emerge from a foundation of respect for human dignity and rights. This balance requires ongoing dialogue and collaboration across disciplines. Scientists, ethicists, policymakers, and community representatives must engage in thoughtful discussions that acknowledge the complexities of stem cell research. For instance, how can we ensure that the benefits of stem cell therapies are accessible to all, rather than a privilege reserved for a select few? This question resonates deeply in a society striving for equity and justice in healthcare. Global perspectives on stem cell ethics offer a fascinating lens through which to examine these debates. Different countries approach stem cell research with varying degrees of openness and regulation. In some nations, the use of embryonic stem cells is embraced and supported, while in others, strict regulations may limit research opportunities. For instance, countries with robust funding and legal frameworks for stem cell research often lead the way in innovation, fostering environments where scientific exploration flourishes. Imagine the vibrant landscapes of research laboratories in these nations, alive with the energy of discovery and collaboration. Conversely, nations with restrictive policies may find themselves at a crossroads, where ethical concerns overshadow the potential benefits. This divergence raises critical questions. How do we create an international dialogue that respects cultural differences while promoting the advancement of science? How can we ensure that ethical considerations do not hinder progress in a field that holds so much promise for humanity? Prominent figures in the field of bioethics contribute significantly to these discussions, offering insights that illuminate the path forward. One such voice, renowned bioethicist Peter Singer, has articulated the need for a careful examination of the moral dimensions of research. As he stated, the ethical implications of our decisions must be at the forefront of our minds as we navigate the uncharted waters of scientific discovery. This call to action serves as a reminder that the intersection of science and ethics demands our attention and commitment. As we explore the ethical landscape of stem cell research, it becomes clear that the journey is as much about the choices we make today as it is about the discoveries we hope to achieve tomorrow. The potential of stem cells to transform medicine is undeniable, yet the ethical considerations surrounding their use require thoughtful deliberation. Each decision we make reverberates through the lives of individuals, families, and communities, shaping the future of healthcare and the very fabric of our society. In this delicate balance, the responsibility of the scientific community is immense. Researchers must not only strive for breakthroughs, but also engage in transparent discussions that consider the broader implications of their work. How do we foster a culture of ethical inquiry that encourages innovation while remaining grounded in respect for human life? This question challenges us to think deeply about our values and priorities as we advance in our understanding of stem cells and their potential applications. The landscape of stem cell research is continually evolving, shaped by advancements in science, societal values, and ethical considerations. As we move forward, we must remain vigilant, ensuring that our pursuit of knowledge does not eclipse our commitment to ethical principles. Imagine a future where the promise of stem cell therapies is realized, not just through scientific achievement, but also through a foundation of ethical integrity and respect for all. This vision calls us to engage in ongoing conversations, to listen to diverse perspectives, and to strive for a world where science and ethics walk hand in hand, illuminating the path toward healing and hope. As we contemplate the journey of stem cell research, we are reminded that the choices we make today will shape the landscape of tomorrow. The ethical considerations we navigate are not just academic debates. They are deeply personal, touching the lives of individuals and families who seek healing and renewal. In this complex interplay of science and ethics, we find the essence of what it means to be human, to seek understanding, to heal, and to honor the dignity of life in all its forms. The road ahead may be fraught with challenges, but it is also rich with possibility, inviting us to explore the depths of our understanding and the heights of our aspirations. The ethical debates surrounding embryonic stem cell use extend into the realm of public policy and societal values, where the implications of scientific advancements often clash with deeply held beliefs. This tension can be seen vividly in various legislative actions across the globe. Some governments have embraced a more permissive stance, recognizing the potential for significant medical breakthroughs, while others impose strict regulations rooted in ethical concerns about the status of the embryo. This divergence in policy raises a critical question: how do we reconcile the advancement of science with the diverse ethical frameworks that govern our societies? Consider the case of countries like the United States, where state-level decisions lead to a patchwork of regulations regarding stem cell research. Some states actively fund and promote research initiatives, fostering an environment ripe for innovation. In contrast, others impose restrictions that can stifle scientific progress. This inconsistency can create a sense of urgency for researchers who may feel compelled to relocate to more supportive environments, seeking the freedom to explore the full potential of stem cells. Ethical discussions are further enriched by the perspectives of individuals who have personally experienced the impact of stem cell therapies. Imagine a parent whose child has been diagnosed with a debilitating condition. The hope that stem cell research could provide a cure brings forth not only excitement, but also ethical dilemmas. Should the potential benefits outweigh the moral concerns surrounding embryonic sources? These personal narratives infuse the ethical discourse with urgency, reminding us that behind every study lies a human story, a life intertwined with the possibilities of healing. Informed consent, as a cornerstone of ethical research practices, takes on heightened significance in the context of stem cell studies. Each participant's decision to engage in research must be respected, ensuring they are fully aware of the implications of their involvement. Yet, how do we ensure that consent is truly informed, especially in complex fields like stem cell research? Researchers must strive to communicate the intricacies of their work in ways that are accessible and comprehensible, fostering a foundation of trust between scientists and participants. Furthermore, the concept of consent extends beyond the individual. As we contemplate advancements in stem cell therapies, it is essential to consider the collective implications for communities. How do cultural beliefs and societal norms shape perceptions of consent? In some cultures, decision-making may be viewed through the lens of collective values rather than individual autonomy. Engaging with these diverse perspectives is crucial in developing ethical frameworks that respect and honor varying cultural contexts while advancing scientific knowledge. As innovation in stem cell research accelerates, the ethical responsibilities of researchers grow increasingly complex. It is not enough to simply generate new therapies. Scientists must also consider their implications for society. Balancing the excitement of innovation with the weight of ethical responsibility is a nuanced endeavor. How can we ensure that the benefits of stem cell research are equitably distributed, rather than becoming a privilege reserved for those with access to resources? The integration of ethics into the fabric of scientific inquiry is essential for fostering a culture of responsibility. Researchers, policymakers, and ethicists must collaborate to establish guidelines that prioritize both innovation and ethical integrity. Imagine a world where breakthroughs in stem cell therapies are accompanied by comprehensive discussions about their societal implications, fostering a holistic approach to scientific advancement. This vision requires ongoing dialogue, where diverse voices contribute to shaping the ethical landscape. Global perspectives on stem cell ethics provide a rich tapestry of insights, illustrating the varied approaches that different countries adopt. In some nations, stem cell research is seen as a moral imperative, a means to alleviate suffering through innovative therapies. Conversely, other countries may adopt a more cautious stance, prioritizing ethical considerations above all else. This divergence raises important questions about how we can foster international collaboration while respecting cultural differences. Prominent figures in the field of bioethics advocate for a balanced approach, emphasizing the importance of dialogue and understanding. Renowned ethicist Françoise Baylis once remarked, "Ethical reflection is a necessary part of the process of scientific discovery." This perspective underscores the need for ongoing conversations that bring together scientists, ethicists, and the public, allowing for a more nuanced understanding of the ethical implications of stem cell research. As we reflect on these global perspectives, we must also consider the impact of public opinion on research endeavors. Public sentiment can influence funding decisions, regulatory frameworks, and the ethical discourse surrounding stem cell research. How do we engage communities in discussions about stem cell ethics? The answer lies in fostering transparency and inclusivity, allowing diverse voices to be heard. In many ways, the ethical landscape of stem cell research mirrors the complexities of human experience itself. Each decision, each debate, is intertwined with the hopes and fears of individuals seeking healing and understanding. The journey of stem cell research is not merely a scientific endeavor, it is a deeply human one, calling us to reflect on our values and aspirations as we navigate the uncharted waters of medical innovation. As we continue to explore the ethical considerations in stem cell research, it is essential to acknowledge that the path forward is not devoid of challenges. Yet it is precisely through these challenges that we can cultivate a deeper understanding of our ethical responsibilities. The fusion of science and ethics invites us to engage in meaningful conversations that honor the dignity of life, while pushing the boundaries of what is possible. In this delicate balance between hope and responsibility, the future of stem cell research beckons us to remain vigilant. Our pursuit of knowledge and healing must be guided by a commitment to ethical principles, ensuring that the benefits of scientific advancements are shared equitably. The story of stem cell research is still being written, and as we pen the next chapters, let us do so with a profound respect for the lives it may touch and the ethical considerations it entails. Chapter 9. The Future of Stem Cell Research and Applications. As we gaze into the horizon of stem cell research, an array of emerging trends begins to unfurl before us, painting a picture of hope and innovation. The landscape is dynamic, teeming with possibilities that promise to reshape our understanding of health and healing. Imagine a world where the boundaries of medical treatment are continuously pushed, where the potential of the human body is no longer limited by age or disease. This vision is increasingly becoming a reality, driven by advances in stem cell technology and therapy. One of the most promising trends is the refinement of techniques to reprogram adult cells into induced pluripotent stem cells. By transforming ordinary cells into stem cells capable of developing into any cell type, researchers are opening new avenues for personalized medicine. Picture a scenario where a patient's own cells are used to generate healthy tissues or organs, eliminating the risks of rejection and ethical dilemmas associated with embryonic stem cells. These induced pluripotent stem cells, or iPSCs, possess the extraordinary ability to differentiate into neurons, heart cells, or even pancreatic cells, offering hope for conditions such as diabetes or neurodegenerative diseases. Moreover, the field is witnessing a surge in the development of organoids, miniature, simplified versions of organs created from stem cells. These organoids serve as powerful models for studying diseases, drug testing, and even personalized treatment strategies. Imagine tiny brains, livers, or kidneys cultivated in a laboratory, providing invaluable insights into how diseases manifest and respond to therapies. This emerging trend not only enhances our understanding of human biology, but also paves the way for more effective treatments tailored to individual patients. As we contemplate the future, potential breakthroughs on the horizon shimmer with promise. Researchers are diligently exploring the possibilities of using stem cells to repair damaged tissues and organs in ways previously thought unimaginable. For instance, the prospect of developing a functional heart or pancreas from stem cells is no longer confined to the realm of science fiction. In recent studies, scientists have successfully engineered heart tissues from stem cells, demonstrating the potential to address heart disease, a leading cause of mortality worldwide. Envision the day when heart transplants become a thing of the past, replaced by regenerated tissues that function seamlessly within the body. Another exciting area of exploration is the use of stem cells in gene therapy. By combining the regenerative capabilities of stem cells with advanced genetic engineering, researchers are working to correct genetic defects at the source. The application of CRISPR technology in this realm has opened doors to unprecedented possibilities. Imagine being able to alter the genetic code of a person suffering from a hereditary disease, effectively eradicating the condition before it even manifests. This harmonious blend of stem cell therapy and gene editing could redefine the landscape of treatment for genetic disorders, offering hope to countless individuals and families. As we stand on the brink of these breakthroughs, the integration of artificial intelligence into stem cell research is heralding a new era of discovery. AI is transforming the way scientists analyze data, design experiments, and even predict outcomes. With its ability to process vast amounts of information far beyond human capabilities, I can identify patterns and correlations that may remain hidden to researchers. Imagine a powerful assistant that can sift through mountains of genetic data, pinpointing the most promising candidates for stem cell therapies. This synergy between human ingenuity and artificial intelligence is poised to accelerate the pace of discovery, ushering in innovations that could revolutionize medicine. One of the most compelling applications of AI in stem cell research is the optimization of differentiation protocols. By utilizing machine learning algorithms, researchers can refine the conditions under which stem cells are coaxed into becoming specific cell types. This meticulous approach enhances the efficiency and yield of desired cells, making therapies more accessible and effective. Picture a future where the process of generating neurons for treating neurodegenerative diseases is as streamlined as programming a computer, reducing the time and resources needed to bring these therapies to fruition. As we envision the next decade, the potential applications of stem cells seem boundless. The vision extends beyond individual therapies to encompass comprehensive treatment paradigms that integrate stem cell technology with holistic care. Imagine healthcare systems where regenerative medicine is a standard practice, where patients are not merely treated for symptoms but are offered solutions that restore their health at the cellular level. This paradigm shift could redefine our approach to chronic diseases, enabling the body to heal itself through the power of its own biology. Furthermore, the future of stem cell research holds the promise of addressing health disparities that plague communities around the world. With advancements in technology and a commitment to equitable access, the fruits of stem cell research could reach underserved populations, offering treatments that were once the privilege of the few. Envision a healthcare landscape where every individual, regardless of their background, has access to cutting-edge therapies that harness the restorative power of stem cells. In this unfolding narrative, ethical considerations remain paramount. As we navigate the potential of stem cell applications, it is essential to maintain a dialogue that respects the moral implications of our choices. The power to heal comes with the responsibility to ensure that advancements are made with integrity and respect for human dignity. This dialogue must include diverse perspectives, fostering an environment where innovation and ethical stewardship coexist harmoniously. As we look forward, the future of stem cell research is illuminated by the brilliance of human creativity and determination. The possibilities stretch out before us like a vast horizon, inviting exploration and discovery. With each breakthrough, we inch closer to a world where the potential of our biology is fully realized, where healing is not just a dream but a tangible reality within our grasp. The journey ahead is not just about scientific advancement. It is a testament to our unwavering commitment to improving lives and shaping a healthier future for generations to come. The horizon of stem cell research is not only illuminated by the advancements in cellular manipulation, but is also enriched by the exploration of new applications that promise to redefine healthcare in the coming decade. Researchers are beginning to envision the role of stem cells in combating some of the most pressing health challenges we face today, such as aging-related diseases. Picture a future where age-related decline can be mitigated by rejuvenating tissues and organs through stem cell therapy. Imagine the potential for improving the quality of life for the elderly, allowing them to lead more active and fulfilling lives, free from the constraints of degenerative conditions. One particularly exciting area of research involves the interplay between stem cells and the aging process. Scientists are investigating how stem cells can be harnessed to rejuvenate aged tissues. This exploration is inspired by the observation that stem cells in older individuals often exhibit diminished regenerative capabilities. The question arises, can we enhance the function of these cells or even rejuvenate them to restore their youthful vigor? Recent studies suggest that certain factors can reactivate the regenerative potential of aged stem cells, hinting at the possibility of therapies that could reverse some aspects of aging. Imagine a therapy that could turn back the clock on cellular function, allowing the body to heal itself as it did in youth. Additionally, the realm of immunotherapy is being transformed through the lens of stem cell research. The immune system, a complex network of cells and molecules, is pivotal in fighting diseases, including cancer. Researchers are investigating how stem cells can be utilized to enhance immune responses, creating more effective treatments for cancer patients. Envision a future where a patient's own stem cells are engineered to recognize and attack cancer cells, thereby providing a personalized approach to cancer treatment. The potential of combining stem cell technology with immunotherapy could lead to breakthroughs that transform oncology, reducing the reliance on traditional therapies that often come with significant side effects. As we explore these emerging trends, we must also consider the integration of artificial intelligence within the broader landscape of stem cell research. The use of AI is not merely about speed. It's about enhancing the precision and effectiveness of experiments. By employing machine learning algorithms, researchers can analyze vast datasets generated from stem cell experiments. This new capability allows for the identification of the most promising therapeutic candidates more efficiently than traditional methods. Imagine a laboratory where a computer can predict the outcomes of stem cell differentiation based on historical data, guiding scientists toward the most effective protocols for generating specific cell types. This synergy could significantly accelerate the path from bench to bedside. Moreover, the application of AI extends beyond data analysis. It also paves the way for innovative biomanufacturing processes. Currently, the production of stem cell-derived therapies is often hampered by scalability challenges. However, by utilizing AI-driven automation, researchers can optimize the conditions needed for large-scale production of stem cells, ensuring that therapies become more widely available. Picture a future where regenerative therapies are produced at a scale that meets the global demand, allowing for equitable access to life-changing treatments. As we contemplate the vision for stem cell applications over the next decade, it becomes clear that the integration of these technologies will shape not only how we treat diseases, but also how we understand health and wellness. The concept of preventative medicine is gaining traction, as stem cell therapies may soon be employed not just to treat existing conditions, but to prevent them altogether. Imagine a healthcare system where individuals are proactively treated at the cellular level, using stem cell therapies to maintain health and vitality throughout their lives. An intriguing vision emerges when we consider the potential for stem cell banking. The idea of preserving stem cells at a young age for future therapeutic use is becoming increasingly feasible. This advancement could revolutionize personalized medicine, allowing individuals to access their own genetically matched stem cells for future treatments. Visualize a world where every person has a personalized repository of stem cells, ready to be utilized in the event of injury or illness, enhancing the body's innate healing process. However, with these advancements come responsibilities. The ethical landscape surrounding stem cell research will continue to evolve, necessitating ongoing discussions about consent, accessibility, and equity. As we embrace the future of stem cell applications, we must ensure that the benefits of these breakthroughs are shared broadly, transcending socioeconomic barriers. The vision is clear, a future where innovative stem cell therapies are not a luxury, but a standard of care available to all. The next decade holds the promise of a transformative shift in healthcare, where the synergy of stem cell technology and artificial intelligence will redefine our approach to medicine. Imagine a world where the healing powers of the human body are fully harnessed, where diseases that once seemed insurmountable can be managed, or even eradicated, through the remarkable potential of stem cells. As researchers and innovators continue to push the boundaries of what is possible, we stand on the cusp of a new era in medical science, one that prioritizes healing, regeneration, and the well-being of all humanity. The journey is just beginning, and the future is bright with possibilities, beckoning us forward into uncharted territories of discovery and hope. Chapter 10: Public Perception and Education on Stem Cells. In the realm of science, the power of understanding is paramount, especially when it comes to the complex world of stem cells. The importance of public understanding of stem cell science cannot be overstated. It is essential for fostering informed discussions about the potential and ethical considerations of stem cell research. Imagine a community where everyone is well informed about the capabilities of stem cells, the potential to heal, regenerate, and transform lives. This understanding can empower individuals to engage with the science, advocate for policy changes, and support funding initiatives that advance research. Yet, public perception of stem cells is often clouded by misconceptions and fears. A lack of education can lead to misunderstandings about what stem cells are and what they can do. For instance, many people associate stem cells primarily with controversies surrounding embryonic stem cell research, neglecting the vast potential found in adult stem cells and induced pluripotent stem cells. There can be a fear that stem cells are synonymous with cloning or unethical practices, which further complicates the public's relationship with this vital field of study. To bridge this gap, effective strategies for stem cell education are essential. First, it is crucial to present information in a clear, accessible manner. Visual aids can play a significant role. Imagine infographics illustrating the journey of a stem cell from its origin to its potential applications in medicine, showing the stages of differentiation and the diverse cell types that can arise from a single stem cell. These visual representations can demystify the science, making it approachable for a broader audience. Engaging storytelling is another powerful tool in education. By sharing personal stories of individuals who have benefited from stem cell therapies, educators can create emotional connections that resonate with the audience. Consider a narrative where a child with a rare genetic disorder receives treatment through stem cells, leading to remarkable improvements in health. Such stories can shift perceptions, transforming abstract concepts into relatable human experiences. Another vital aspect of education is fostering dialogue. Public forums, workshops, and community discussions can provide platforms for individuals to ask questions, express concerns, and engage with experts in the field. Imagine a community gathering where researchers and ethicists come together to discuss the advancements and ethical considerations surrounding stem cell research. This collaborative approach can nurture a sense of trust and transparency, allowing the public to feel involved in the scientific process. Addressing misconceptions directly is also crucial. For instance, many people fear that stem cell research is an unregulated field, fraught with risks. In reality, rigorous ethical guidelines and oversight govern research practices. By educating the public about these safeguards, we can alleviate fears and foster a more informed perspective. Advocacy plays a fundamental role in shaping stem cell policy and funding. Organizations dedicated to advancing stem cell research work tirelessly to create awareness and support. Advocacy efforts can mobilize communities, encouraging them to participate in campaigns that push for increased funding and more inclusive policies. Imagine a nationwide movement where individuals, scientists, and advocates come together, united by a shared vision of harnessing stem cells for the greater good. The voices of those affected by diseases that could benefit from stem cell research can amplify the call for action, bridging the gap between scientific possibility and public policy. Prominent figures in the scientific community often emphasize the necessity of advocacy. As one influential researcher noted, science thrives in an environment of open dialogue and collaboration. This sentiment underscores the importance of creating a supportive atmosphere where science can flourish, driven by informed public engagement. Moreover, collaboration between scientists and educators can lead to innovative educational programs. Schools and universities can integrate stem cell science into their curricula, sparking curiosity among students. Imagine young minds exploring the intricacies of cellular biology, equipped with the knowledge that these concepts can lead to groundbreaking advancements in medicine. By cultivating interest from an early age, we can prepare the next generation of scientists and informed citizens, ready to contribute to the field. In addressing fears, it is also essential to highlight the positive impacts of stem cell research. Public awareness campaigns can focus on success stories, showcasing how stem cells are being used to treat conditions such as spinal cord injuries, degenerative diseases, and even certain types of cancer. The transformative potential of stem cells can shift the narrative from fear to hope, igniting a passion for exploration and understanding. As we reflect on the public perception of stem cells, it becomes evident that education is not merely a tool, it is a bridge to understanding. The more the public knows, the more they can engage with the science and its implications. The relationship between science and society is symbiotic. As public understanding grows, so too does support for research and innovation. In summary, the journey of public understanding of stem cell science is ongoing. Through effective education, open dialogue, and advocacy, we can cultivate a society that not only understands the science but embraces the possibilities it presents. The power of stem cells lies not just in their biological potential, but in the collective vision of what we can achieve together, fostering a future where healing and innovation are within reach. The journey toward a well-informed public on the topic of stem cells is a continuous one, requiring dedication and creativity. One of the most effective strategies for enhancing public understanding is to incorporate interactive learning experiences. Imagine a science fair where children and adults alike can engage with hands-on exhibits, observing stem cells under microscopes or participating in simple experiments that illustrate cellular processes. These immersive experiences can spark curiosity and foster a deeper appreciation for the science behind stem cells, transcending traditional classroom learning. Moreover, the integration of technology and education can significantly amplify outreach efforts. Online platforms and social media can serve as powerful tools for disseminating information. Consider the impact of a viral video that explains stem cell therapy through compelling visuals and relatable narratives. By harnessing the power of storytelling within digital media, we can reach diverse audiences, breaking down the barriers of misinformation and fear. Social media campaigns can highlight not only the science, but also the human stories that underpin the research, creating a more personal connection to the topic. As we think about addressing misconceptions, it is vital to recognize the role of trusted voices in the community. Scientists, educators, and healthcare professionals can serve as ambassadors of knowledge, providing credible information and countering myths surrounding stem cell research. Imagine a local doctor who takes the time to explain the facts about stem cells to patients, dispelling fears and misconceptions with empathy and clarity. This kind of grassroots education can build trust and encourage more informed discussions about the topic, empowering individuals to advocate for research and policy changes. Furthermore, the impact of renowned figures in the scientific community cannot be underestimated. When respected scientists share their insights and experiences, they can inspire public confidence in stem cell research. Take, for example, the efforts of Dr. Shinya Yamanaka, who was awarded the Nobel Prize for his pioneering work in creating induced pluripotent stem cells. His journey, marked by both challenges and triumphs, serves as a beacon of hope, illustrating not only the potential of stem cells, but also the dedication required to explore their possibilities. When scientists share their passion and commitment, it resonates with the public, transforming abstract scientific concepts into relatable human endeavors. In the realm of education, it is also essential to address ethical considerations transparently. By engaging in open conversations about the ethical implications of stem cell research, we foster a culture of respect and understanding. Imagine a community forum where ethical dilemmas are discussed openly, allowing individuals to voice their concerns and learn from experts. This engagement encourages critical thinking and nurtures a balanced perspective on the potential benefits and risks associated with stem cell research. Advocacy plays a pivotal role in shaping public perception and policy surrounding stem cells. Organizations dedicated to stem cell research can mobilize communities, leveraging the collective voices of individuals affected by diseases that may benefit from stem cell therapies. Picture a campaign where patients, families, and advocates come together to share their stories, illustrating the urgent need for funding and support for research. Such advocacy not only raises awareness, but also demonstrates the human impact of scientific advancements, making the case for increased investment in stem cell research. Moreover, collaborative efforts between advocacy groups and educational institutions can enhance outreach initiatives. By partnering with schools and universities, organizations can create programs that introduce students to the world of stem cells from an early age. Imagine high school students conducting research projects on stem cells, guided by experienced mentors in the field. This hands-on experience not only cultivates scientific literacy, but also inspires the next generation to pursue careers in biomedical research. In addition to grassroots efforts, the influence of policy at national and international levels shapes the landscape of stem cell research. Legislation can either promote or hinder advancements in the field, making it crucial for advocates to engage with policymakers. Imagine a scenario where a coalition of scientists and patient advocates meets with lawmakers to discuss the potential for stem cell therapies to address pressing health challenges. By presenting compelling evidence and personal stories, they can influence policy decisions that prioritize funding and support for research initiatives. As we navigate the complexities of public perception and education on stem cells, it becomes clear that fostering a culture of understanding requires a multifaceted approach. By embracing innovative educational strategies, leveraging technology, and engaging trusted voices, we can dismantle barriers to understanding and cultivate an informed society. The interplay between advocacy and education creates a powerful force, driving progress in stem cell research, policy, and ultimately, patient care. In the grand tapestry of science, the potential of stem cells is a vibrant thread, woven together by the collective efforts of researchers, educators, advocates, and the public. Every story shared, every question answered, and every myth dispelled contributes to a greater understanding of the science that holds the promise of healing. The journey toward a well-informed populace is ongoing, but with each step taken, we move closer to a future where the potential of stem cells is recognized, embraced, and harnessed for the betterment of humanity. As we envision this future, we must remain committed to education, advocacy, and open dialogue, ensuring that the conversations surrounding stem cells are not only informed, but also infused with hope and possibility. The journey toward public understanding of stem cell science is not merely an academic endeavor. It is a collective mission that requires the engagement of diverse voices. As we explore the role of advocacy in shaping stem cell policy and funding, we begin to see how collaboration can amplify efforts and create meaningful change. Advocacy organizations play a vital role in uniting patients, families, and scientists under a common purpose, fostering a grassroots movement that can influence decision-makers. Imagine a scenario where individuals impacted by diseases such as multiple sclerosis gather to share their experiences. Their stories, woven together, create a powerful narrative that captures the urgency for research funding. These personal accounts can resonate deeply with policymakers, prompting them to support initiatives that might have once seemed distant or abstract. As these advocates speak out, they illuminate the direct connection between scientific research and the potential for life-changing therapies. One of the most compelling aspects of advocacy is its ability to humanize scientific endeavors. When individuals affected by debilitating conditions advocate for stem cell research, they bring a face to the statistics and data that often dominate scientific discussions. This shift from abstract numbers to personal stories can transform the conversation, fostering empathy and understanding. Advocates help bridge the gap between the lab and the lives of those in need, reminding us that behind every breakthrough is a community hopeful for progress. In this journey, education remains a cornerstone of advocacy. By equipping advocates with accurate information about stem cells and their potential, we empower them to articulate their message more effectively. Imagine workshops where community members learn about the science of stem cells, the latest research, and the ethical considerations involved. With this knowledge, they can engage in informed conversations, dispelling myths and addressing concerns within their communities. Furthermore, the integration of patient voices into the research process enhances the relevance of scientific inquiries. When researchers collaborate with advocates, they gain insights into the real-world challenges faced by patients. This partnership can lead to more targeted research efforts, ensuring that the scientific community addresses the most pressing needs. For instance, understanding the specific challenges faced by patients with spinal cord injuries can guide researchers in developing therapies that truly make a difference in their lives. The landscape of stem cell research is shaped not only by scientific innovation, but also by the policies that govern it. Advocacy efforts can significantly influence funding decisions at both national and international levels. As we envision a future where stem cell therapies are accessible to all, we must recognize the importance of sustained advocacy to ensure that research is prioritized. Consider the impact of concerted lobbying efforts that seek to secure government funding for stem cell research initiatives. By presenting compelling evidence of the potential benefits, advocates can persuade lawmakers to allocate resources that support ongoing studies and clinical trials. This collaborative approach, bringing together scientists, patients, and policymakers, fosters an environment where research can thrive. Public perception is further shaped by the media's portrayal of stem cell science. Advocacy groups can work to provide accurate information to journalists, ensuring that the narrative surrounding stem cells is grounded in facts. When the media highlights success stories, it can inspire hope and encourage public support for research. Imagine a news segment featuring a patient who has regained mobility through innovative stem cell therapies, showcasing the tangible benefits of scientific advancements. Such stories can galvanize community support and influence funding decisions. As we reflect on the importance of public understanding and advocacy in the realm of stem cell research, it becomes evident that we are part of a larger narrative, one that encompasses hope, healing, and the relentless pursuit of knowledge. The interplay between science, society, and policy creates a rich tapestry that can lead to transformative outcomes. By fostering a culture of education, collaboration, and advocacy, we can pave the way for a future where the potential of stem cells is fully realized, benefiting generations to come. As we contemplate the journey ahead, we must remain committed to nurturing informed discussions and empowering individuals to advocate for change. The power of stem cells lies not only in their scientific promise, but also in the collective voices that champion their potential, ensuring that the dialogue surrounding stem cell research is one filled with possibility and hope.